So, hello students, welcome to your own channel Vedantu NEET English. This is Basavaraj Sir, your biology master teacher students. Now. In yesterday's class, Gopika ma'am solved a lot of questions from your zoology. In today's class, we'll be doing from botany. And let me tell you, in today's class, the majority of the questions, almost all the questions will be from your class 11th. Sorry, mention any students. So, just saying before we start, this is going to be a complete class 11th lecture, right? So all the way from your living world to the entire plant physiology, every single chapter, morphology, anatomy, cell cycle, biomolecules, every single chapter from your class 11 botany. And let me tell you why this session right now, when we could be doing a long marathon of your class 11 or class 12th. The reason we are doing this session right now is to check your knowledge and how much of preparation is going on. For example, students, if you are able to solve almost, let's say, 80% or 90% of these questions in today's class, 80%, 90% of preparation is done. You're like, okay, sir, I am very confident in biology. I'm very confident in botany and I can score out of 4. Okay, students. So, students, if I'm audible to all of you, if I'm visible to all of you, quickly some fire emoji in the chat to let me know, yes, sir, I am audible and visible to all of you. Students, quickly in the chat. Students, quickly. And before we start, yes, hello, good morning, students. Lamb of God, praise Lord. Okay, good morning to all of you. Okay, now I want everyone to rate your botany preparation out of 10. All of you in the chat, quickly. I want everyone to rate your botany preparation, sir. Out of 10, my botany preparation is around, let's say, 7, 8, 9 or 10 on 10. In the chat, quickly, students. In the chat, what is your botany preparation right now? And all of you students, before we start the class, I want everyone to quickly go and get your bottle for all of you. All of you quickly go and get a water bottle because it's going to be a very long class. Yes, it will be mentally draining. So I want everyone to get a water bottle. Get your mind here, okay? And start solving questions. Any question you have a doubt, you can always put it up in the chat, okay? Minus students. Yes, today is the second PUC result. I'm going to check that again. Yes, 7 out of 10, 8 out of 10. Minus 555. Students, come on. Botany is very easy. Botany is very easy. And I'll prove it to you again today. Okay, so with that same energy and same enthusiasm, let's start today's class, students. Can we start? Yes, with same energy and enthusiasm. Let's start with the first question on the board. So we'll be starting off with the living world. Eventually, as we go ahead, we'll go to the next chapter one by one. And students, if you want to know where can we get all of these notes, all of these notes is going to be available on the Telegram channel. Basavaraj Sir Biology. You can go here and get all the notes possible. Okay. Now, students, the first question from living world, obviously, it's going to be a much more easier question. Very laddu question. I call them laddu questions. Okay, very easy laddu question. The first question on the board. Okay, the first question on the board for all of you. Which of the following is against the rule of ICBN? Against the rule of ICBN. It's like no. Against the rule of ICBN. Handwritten scientific name should be underlined. Yes, makes sense. Every species should have a generic name and a specific epithet. Yes, absolutely correct. Scientific names are in Latin or should be italicized. Yes. Generic name and species name should be written in starting with small letter, students. Do you think so? Do you think so, students? The first letter and last letter, right? First letter and last both should be small letter? No. Because, students, if you know that starting generic name should be in what? Should be in capital. It should be in capital letters. For example, students. For example, let's take. One second, students. What happened here? Oops. For example, if you go here, the example of Mangifera indica, you can clearly see in Mangifera indica, the M is going to be capital and I is going to be small. So, generic name has to be in capital, rest everything can be in small. Very easy question. Okay, very easy question. Let's go to the next question, students. Let's go to the next question. Students, can all of you keep a track of your marks? Is it possible? D for Diksha sir, yes. Can all of you keep a track of your marks? See, every right answer 1 mark. Just keep a track of every right answer 1 mark. No negative marking in my class today. Free of all of you. No negative marking. Every right answer 1 mark. Keep a track of your marks. It'll be very easy. Okay. Next question on the board, students. Next question on the board from NEET 2021. Which of the following belongs to the family Muscidae? Family Muscidae, students. Now again, very important, students. Very important, right? Because there's a table, all of you remember the table, right? A table with all the, for humans, for mango, for housefly, and as well as for your wheat. Triticum aestivum. So the table is there. In the table, they can ask you anything. They can ask you family, order, or they can ask you scientific name can be asked. Okay. So what is the answer, students? Answer is going to be housefly, that is Muscidae. Here is the table which I am talking about. Here they can ask you any of the following. Okay. Shall we play a small game, students? Shall we play a small game? Okay. The small game is very simple game. Okay. Now I'll give you exactly 10 seconds. I'll give you 10 seconds to look at this table. I want everyone to just keep staring at the table. Okay. Keep staring at the table. And after 10 seconds, I will ask you one question. Okay. Let's start. Your time starts now. Go. Friends, your time starts now. Go. Tell me once you're done, just say done, sir. Okay. Your time is starting now. Let's just go. 5, 1. Time's up. Okay. Students, can anyone tell me in the chat what is the family of mango plant? The family of mango. Quick. The family of mango. And the second question is, what is the order of housefly? What is the order of housefly and the family of mango? Quick answers I want. And these are free questions. You can't get marks for this. Okay. You can't get marks for this. Okay. So the family is Anacardiaceae for your mango, and Diptera is the order for the housefly. So these are the small things which they can ask you in examination from the living world. Apart from the students, there is one more small topic which you can study. Let me show you. Let me show you, students. This is the question which you can get here. This is the only question which you can get here. In the taxonomical categories, which hierarchical arrangement is in ascending order is correct in the case of animals? In the case of animals means there will be phylum. Yes, there will be phylum. So what are the trick here, students? The trick is very simple here. Right? Keep pots clean or family gets sick. Keep pots clean. Keep pots clean or family gets sick. Very simple. Option number 2. Option number 2, students. Option number 2. So if you're able to keep a track of your marks, very good in the chat, all of you. That's going to be option number 2. So they can ask you this entire thing here. Entire thing they can ask you. Okay. Now here the only question they can ask you is similarities. For an example, students, tell me. Let's say we belong to which kingdom? Can anyone tell me we belong to which kingdom? We belong to kingdom Animalia. Okay. What about sponges? Sponges also belong to kingdom Animalia. Now you tell me, students, do humans and sponges, do we have similarities in us? No. So as we go from, students, remember, all of you remember, students, in the chat, at kingdom level, very less similarities are observed. As we go down and down and down, similarities will increase. Similarities will increase. In the kingdom level, very less similarities. At species level, we have more similarities. This is the only question which you need to worry here. Done. Very simple, student. Very simple. Okay. Look at this question again. Binomial nomenclature. They can ask you such question one more time. They can ask you such question one more time, students. They can ask you such question one more time. Very easy question. X and Y are the components of binomial nomenclature. Binomial nomenclature, students. This naming system was proposed by Z. It was proposed by Carl Linnaeus. So these two are gone. So between these two, students, you should tell me which is X and which is Y. So first, X is the first letter, right? X is the first one. That is going to be what? That is going to be generic name or the genus name. And second is going to be species epithet. It's going to be species or specific epithet. Okay. So option number 1. So all of you students, can anyone tell me what is your marks? Yeah, I'm feeling a lot better now because students, I've been taking a lot of medication, a lot of painkillers and everything. So small physiotherapy also. Yes, A for Basavaraj Sir. How is A for Basavaraj Sir? Okay. Done, students. Option number A. Okay. Can I see the marks? Students, this is very easy again. House belongs to Muscidae. Muscidae, easy. Okay. So before we go into plant kingdom, students, before we go to plant kingdom, can I see the marks in the chat, all of you? Before we go to the plant kingdom, can I see the marks in the chat? Now, can everyone type in the chat right now? Living world, done and dusted. That's all. Living world, done and dusted in the chat, all of you. Hashtag living world, done and dusted. Because apart from that, living world will not ask you a lot of questions. The question will come from the taxonomy, the nomenclature, or the table which I showed you, or the hierarchy. Three concepts, easy. So it's done and dusted right now. Done and dusted. Okay. Now, students, before we start, I want everyone to drink a lot of water. All of you drink some water now. Now, shall we start? Students, shall we start now? Yes. Done and dusted. Now, similarly, every single chapter we'll do done and dusted today. Every single chapter. And in the comment section, students, I want someone to add a timestamp. If anyone can help it out, if no one does, I'll only do it later on. I'll put a timestamp of every single chapter. You can come back here and watch the lecture if you are getting confused in any question in henceforth. Okay. Now, students, let's go with the next concept. That next chapter is going to be plant kingdom. And here is the first question on the board. Here is the first question on the board, students. Okay, first question on the board. Now, from evolutionary point of view, look at evolution. Okay, from evolutionary point of view, retention of female gametophyte being retained with developing young embryo on the parent sporophyte for some time is first observed in. The question here is hinting at something called as seed habitat. Okay. Seed habitat. What is seed habitat, dear students? Where do you see the seed habitat? Anyone? In which of the following condition do we see seed habitat first? Okay. All of you tell me, sir. Very easy question. What is Laddu question here? You can easily tell me, sir. It's going to be option number 4, pteridophytes. Now a question to all of you. Under pteridophytes, is it going to be homospory or heterospory? Where do you notice the first seed habitat, students? All of you. Quickly, students in the chat. Where do you see the first seed habitat, in homospory or heterospory? If you're my student, you can tell this answer very easily. It doesn't take more than 10 seconds. It doesn't take more than 10 seconds. Very easy, very easy. Students, quickly, seed habitat. All of you. Quick. Yes, in the case of heterospory, we see the seed habitat. So, very simple answer, students. If you see here, under heterospory, we can see this. The female, from here it starts, the female gametophyte in these plants are retained on the parent sporophyte for a variable period of time. The development of zygote into young embryo takes place within the female gametophyte. This event is called as seed habitat. It's called as what, students? Seed habitat in heterospory. Okay. So I hope this NCERT snips is making easier for you to, cross check your answers. NCERT snips. Okay. Next question on the board. Pinus seeds cannot germinate and establish without fungal association. This is because students, tell me, why don't think Pinus can survive on its own? Is there a small secret here? Why Pinus needs someone else, some babysitting it needs? Students, tell me in the chat. All of you. Hello. Students, have you liked the video? First of all, tell me in the chat. Students, I want everyone to quickly smash the like button first of all. All of you quickly smash the like button. There's a small target here. Very small target, so I want everyone to quickly smash the like button also. It contains inhibitors and prevent germination. No. The embryo is immature. No. It is an obligate association with mycorrhizae. Absolutely, students. So remember this. In the case of Pinus, it's gonna be mycorrhizae. Okay, MP. Now, what about in the case of Cycas? What exactly happens in the case of Cycas? In the case of Cycas, right? In the case of Cycas, it's called as CCC. What is CC, students? C for Cycas, C for cyanobacteria, C for colloid roots. So in the case of Cycas, it is CCC, cyanobacteria, colloid roots, as well as something called as Cycas. So that is CCC in the case of Cycas. Pinus, it is going to be mycorrhizae. It's going to be mycorrhizae, students. So remember this throughout your life. Okay. So Cycas, small specialized roots called as colloid roots, are associated with nitrogen fixing cyanobacteria. C here. Okay. C for cat. Exactly. C for cat. Okay. C for cat here. Now, next concept on the board. Next question on the board. From NEET 2020. Which of the following statements is, again, they're asking about incorrect statement about gymnosperms. Incorrect statement about gymnosperms, students. Quickly. All of you keep a track of your marks in the chat because it'll be easy for you to see, track your progress also. How many questions you're able to answer, how many questions you're not able to answer, it becomes easier at the end of the day. Okay, C for cash also here. C for cash cat. Okay. Which type of statements is incorrect about gymnosperms? They are heterosporous. Yes, they are heterosporous. Most of them have narrow leaves with thick cuticle. Yes, their seeds are not covered. Yes, they are naked seeds. Male and female gametophyte are in free living, students. Are they free living? Students, tell me, are they free living? Absolutely not. Very good. They're not free living. If you remember, students, this is my notes which I've taught you, life cycle of gymnosperms, monospore development here. Here, if you notice here, students, in the case of gymnosperms, we can see that the female gametophyte is present inside the sporophyte. Female gametophyte is present inside the sporophyte, and the female gametophyte is not independent here. It is dependent on the sporophyte. Female gametophyte is dependent on the sporophyte here. Dependent on the sporophyte. Clear, students? Clear? So here it is not independent. One more question I can ask you, students. Can anyone tell me what is the ploidy of endosperm in gymnosperm? Yeah. What is the ploidy? Exactly, student. Genie, exactly. What is the ploidy of endosperm in the case of gymnosperms? Haploid, diploid, or triploid? Who can tell me? Haploid, diploid, or triploid? Quick answer, students. I don't want anyone to be lazy in my class. Only if you're active, you will learn a lot. Again, students, you know the drill in my class. If you are active, yes, you can. Otherwise, if you're not active, you're lazy. I'm like, sir, why should I answer in your class? You'll not gain knowledge. You'll not make mistakes. Now, this is where I catch hold of all of you. This is where I catch hold of all of you, students. All the students are telling me answer in the chat. Let me show you a small proof which I've taught in the class. Here, students, I've always told you, in the case of your gymnosperms, this is your female gametophyte here. The zygote here. The zygote develops into the embryo here. The zygote develops into embryo, diploid. What exactly happens in the case of endosperm? Endosperm actually develops from the female gametophyte. Female gametophyte develops into endosperm, and the female gametophyte is always haploid. Gametophyte is haploid. Gametophyte is haploid. Sporophyte is diploid. So this is a female gametophyte which is going to be haploid. It develops into endosperm. So endosperm is going to be what, students? Tell me in the chat right now. All of you made mistakes? Huh? Students, all of you made mistakes? Yes? Who made mistakes? Now, students, raise your hands. So in examination, will you make the same mistake again? No. So in the case of gymnosperms, the endosperm is what? It is going to be haploid in nature. It is going to be what? Haploid in nature. In the case of angiosperms, it's going to be triploid in nature. So please do not make the same mistake again in examination. This is why I tell you. This is the prime example why I tell you. If you attend my class, if you participate in the class, right, one by one, if you participate in the class, right, if you make mistakes right now, you don't have to make the mistakes in examination. Now, all of you made the mistake right now. Yes. So you will not repeat in the examination. Done, students? Done? All of you getting the point? Students, here is one more question. I forgot to ask all of you, students. I forgot to all of you. All of you had breakfast, right? Yes, all of you had breakfast. Huh? Eat breakfast because there is no energy. If there is no ATP molecules in your body, you cannot function in my class. Because my class, you need a lot of energy to attend also. Because I will drain all of you. Okay. Now, Floridian starch has structure similar to. First of all, students, tell me in the chat. Floridian starch belongs to which of the algae? Chlorophyce, Pheophyce, Rhodophyce. Quickly in the chat, students, tell me. First of all, tell me, students, in the chat, which of the following classes does you can find Floridian starch? Red, brown, green. Quick, students. That is the answer I want. First of all, you will tell me, sir, Rhodophyce. Yes, it's going to be Rhodophyce. And what is it similar to? The structure is similar, students. The structure is similar to amylopectin and glycogen. If you come to Rhodophyce, NCERT snips here, last line here. The food stored is Floridian starch, which is very similar to amylopectin and glycogen in structure. Okay, students. Can anyone tell me, students, is amylopectin starch? Yes or no? Is amylopectin starch? Yes or no, students? Quickly in the chat. You will tell me, sir, yes, amylopectin is a variant of starch or cellulose. Starch or cellulose, students, quickly. Amylose and amylopectin is a variant of starch or cellulose. Quick. Students, quickly, I want quick answers. Yes, it is. Yes, it is. Okay. Now, students, what is the difference between amylopectin and glycogen? The difference between amylopectin and glycogen, the first difference is glycogen is found in humans, in living animals, while amylopectin is found in the case of plants. Apart from that, students, is there a variation in branching? Yes. Here, alpha 1,6 branching is a lot. Alpha 1,6 branching is little less here. Here, alpha 1,6 branching is more frequent branching can be observed in the case of glycogen. So remember all this. Okay. So I want everyone to keep a track of your marks. Yes. All of you keep a track of your marks. Yes, exactly. Christina, remind them, Christina. Smash the like button already. Walk in. Next question on the board. Strobilus or cone are found in? Strobili or cones are found in. So first of all, students, remember what is a strobili, what is a cone. Understand, both are same first of all, okay. What is strobili? Okay, strobili is nothing but, all of you remember that fertile special leaf? See, if you remember, students, in the case of pteridophytes, leaves help in reproduction. All of you remember, students? Yes or no? Tell me in the chat. Leaves help in reproduction. And there is something called as fertile leaf. The fertile leaf is called as sporophylls. When there is a group of sporophyll, when there are multiple sporophyll and arranged in a compact structure, that will give rise to a cone, and this cone can be found in your equisetum and selaginella. In equisetum and selaginella, we can find the cone. So I will read all of you the inside lines here. See, in selaginella or large macrophylls, the sporophytes bear sporangia that are subtended by leaf-like appendage called as sporophyll. See, sporophyll. In some cases, sporophylls may form distinct compact structure. So multiple sporophyll are joined together to give rise to a cone or strobili. Students, do you realize your concept is also getting revised right now, not just solving the questions? This is the advantage of solving questions. The concept becomes clear again. Okay. Now, all of you keep a track of your marks in the chat. Yes. With that, we come to the next question on the board. Yes. What is the marks? Quickly, students, what is your marks in the chat? 10, no? 10. I don't even know how many questions we did, students. Let me tell you, students, we have more than 250 questions today. Let me tell you one thing. We have more than 250 questions. So, students, how many of you can watch? How many can survive until the very end? Tell me in the chat. How many of you can survive till the very end? I doubt many can survive. I am very sure many students in the midway only be like, sir, nahi hoga. Like, I cannot do it at all. This is not my cup of tea. I cannot solve 250 questions in one day. You have to, students, you have to. Because all the time you wasted should make up for it. Right? So, how many of you ready in the chat for 250 questions today? Yes. So, which of the following pairs are unicellular algae? See, Gelidium, no. Anabina is Volvox, no. Chlorella spirulina, yes. Laminia sargassum, no. So, answer is 3. Students. Okay. Yes, I'll take genetics, students. I will take genetics after this. Okay. Don't worry. Genetics is coming up. Okay. Students, how many of you want genetics? How many of you want to revise, sir, principles of inheritance? I am not able to solve numericals. So, principles of inheritance, I am not able to solve the questions of your color blindness, sickle cell anemia questions. Now, I'm not talking about generic questions. I'm talking about your crossings. How many of you are not able to solve the questions of hemophilia? In the chat, students. Quick, students. How many of you are not able to solve that, sir? I am not able to solve some questions on monohybrid, dihybrid, right? How many of you, students? Do you want one chapter, one proper chapter which will make sure you can solve any question of your genetics? Any question if they give you also, you should be able to solve. Do you want such question? Do you want such session? I'll make it happen, don't worry. Where we'll be doing all the numericals, all possible numericals of your genetics, every single concept, every single crosses which you can expect in an examination, every single crosses. Okay. Now, next question on the board. Oh, student, this is a very easy question. Before I read the question also, you can tell the answer to this question. Genera like Selaginella and Salvinia produces two kinds of spores. Such plants are known as heterospory, students. This doesn't require much thinking, brain capacity. You can tell easily. Okay, heterospory. Now, next question on the board. Now, first of all, students, first of all, yes, plant physiology. If you want plant physiology, we will do one shot plant physiology one more time. What is there in that? We will do plant physiology one more time, one short plant physiology. Proper 5 to 5 hours, 6 hours, full plant physiology will do. Okay. Now, first of all, students, tell me, is GEM part of sexual or asexual reproduction? Tell me in the chat. Sexual or asexual reproduction? GEM, which is it? GEM is part of sexual or asexual reproduction. Quick. Students, quick. Students, quickly smash the like button, all of you. Sir, give mnemonics for example. Okay, Gene, today only I'll tell you. Students, today only I will teach you all possible mnemonics for your morphology also. Today only I'll teach you, don't worry, it's already in mind. Okay, so you will tell me, sir, it is asexual. And where is it found, students? It is found in the case of Markantia. It is found in the case of Markantia. And Markantia is an example of, students, Markantia is an example of liverworts. Liverworts. Simple as that. Liverworts is an example of, Markantia is an example of liverwort. Okay, Markantia. See, students, it's a asexual part of asexual reproduction in liverwort, GEM, asexual bud. Okay, asexual bud. Now, they can ask you one more question. Is GEM unisexual or is it going to be unicellular or multicellular, students? Is GEM going to be unicellular or multicellular? Students, all of you are asking one by one, so do entire syllabus one more time. Students, I want everyone to listen to me very carefully. Okay. In today's comment box, I want everyone to type what exactly you need. Sir, if you want, I can revise the entire genetics. If you want me to revise the plant families, we'll do that. If you want me to revise the entire plant physiology, we'll do that. So let me know in the comment section what you want. You can't be like, sir, do the entire syllabus one more time. Not possible. Okay. So it's going to be multicellular in nature. It's going to be multicellular in nature, students. Multicellular. Now, some of you will tell me again, sir, do the entire ecology one more time. How is it possible to do entire ecology? We can do it, it's not a big deal, but if you want entire ecology, again, let me know in the comment section, because ecology, let me tell students, ecology you can usually get around, let's say 5, 6, 7, 8, 9, 10 to 12 questions easily. In ecology, you can get around 12 questions easily. Okay, so students, quickly. 12 questions, how many marks? 12 questions, how many marks, students? Quick. 12 questions from ecology can easily come. 12 questions from ecology can easily come, and that many marks you will get. Okay, so you can't ignore. Which of the following produces carrageen? Carrageen is part of your red algae. Carrageen is a type of hydrocolloid. Hydrocolloid. What does it do, students? It will help in holding water. That is present in the case of red algae. Wait. I can ask one more question here. Students, tell me in the chat quickly, students. Okay. 48 marks, students. Now, students, you tell me in the chat, can you afford to even miss ecology at this point? If you're able to get right, let's not even say 48, let's say around 44 marks or something, right? 44 marks. Can you afford to lose 44 marks? Yes, ecology is easy. But if you don't revise ecology, and God promise, exam, you will not be able to remember ecology. That is important, ecology is. Okay. Everyone will tell one chapter now. You cannot. Now, students, let me ask all of you one more question here. In which of the following algae do we find algin? See, carrageen in the case of red algae. What about algin? Who can tell me, students? Algin, where do you find? You tell me, sir, in the case of your Phaeophyceae or brown algae. Phaeophyceae or brown algae, we can find algin. Okay, algin. Students, next question on the board. Which of the following algae contains mannitol reserve food material? Again, answer was previous question. Previously, which I gave you the question, same answer is here also. Okay, which of the following algae contains mannitol as reserve food material? Mannitol and laminarin. Mannitol and laminarin is found where? Mannitol and laminarin is found in the case of where? Brown algae again. Phaeophyceae. Okay, but example is given. Example is given, students. Volvox, no. Eulothrix, no. Gracilaria, red. So very easy, students. Very easy. Ectocarpus, exactly. Students, so you need to know the examples here. For example, you see here, Ectocarpus, Dictyota, Laminaria, Sargassum, Fucus. All of these are examples of your brown algae. Similarly, Gracilaria, Gelidium, right? Or Polyporphyra, all the questions examples of your red algae. In the case of green algae, similar examples are there. So learn the examples because they can ask you the questions. You might know the concept, but if you don't remember the examples, RIP. Next question on the board. Next question on the board. Okay. All of you. Nandini is asking plant kingdom one more time. Students, one by one, you tell me, sir, do entire class 11th, do entire class 12th one more time. Sir, you do one marathon where we do 10 hours, you teach the entire class 11th, 10 hours, you do entire class 12th, and we'll be happy at the end of the day. Huh? Look at this question now. Read the following statements and identify the characters related to algae shown in the diagram. First of all, students, first of all, this algae right here is Cara. The algae right here is Cara. So the first thing you need to identify here, the first thing which you need to identify here is going to be Cara belongs to which of the following classes? Is it going to be Chlorophyceae, Fucaceae, Rhodophyceae? So you will tell me, sir, Cara belongs to Chlorophyceae. Cara belongs to Chlorophyceae. Okay, green algae. So Chlorophyceae, yes. Starch, yes. Monoecious plant. I can't teach students. If I tell you the answer, you'll get to know the answer. No, no. Students, is it monoecious? Does it show oogonium and antheridium? Yes, it does. Let me show you, students. Let me show you a diagram for it. Before I go, see this. This is the microscopic diagram of your Cara. And if you can see, students, it has something called as oogonium and antheridium. Oogonium and antheridium is present. Is present, students. It is present here. Okay. So A, B, C is the answer. Okay. A, B, C is the answer. Again, this is not present in your NCERT. It's present, let's say, it was part of your class 12th. How do organisms reproduce before? So, by chance, if they ask you also, you should know. By chance, if they ask you, you should know. Okay? No. Cara. Okay. Crafty. Okay. Next question on the board. Again, students, very easy question. Match the following. Let's see who does it first. If I do it first, tell the answer in the chat. If I do it first, you do the answer. Okay. Let's see who does first. Chlamydomonas is going to be algae. A is going to be 3. Cycus is going to be gymnosperm. B is going to be 4. Selaginella is going to be your pteridophyte heterospore. And sphagnum is going to be a peat moss. D is 1. D is 1. Only one option is there actually. D is 1. Only one option is there, students. Only one option is there. D is 1. Only one option is there. Friends, who did it first? Me or you? I think I did it first now. Students, if you don't, students don't lose to me right now, I'm telling you. Let's see, it's a competition between now. It is not a competition between all of you. All of you are good enough right now to compete with me now. All of you have an equal knowledge of your NCERT as me right now. So it should be a competition. Who can do first now? Can you do first or I do first? Okay. So let's see who does next question first. Okay. So all of you, your marks in the chat. Students, what is your marks in the chat quickly, students? Students, please smash the like button. All of you quickly smash the like button first of all in the chat. Okay. Okay, students, again, competition. The competition is very simple. Who does the answer first? Among you also, who does first, or if I do it first? Okay. Next question on the board. 3, 2, 1, go. Which of the following structures is haploid in ploidy? Primary endosperm news, primary is triploid in nature, primary mesosphere is diploid in nature, protein is going to be haploid in nature, primary endosperm nucellus is going to be diploid in nature, diploid, triploid. So answer is option number 3. Answer is option number 3. Who told first, students? Who told first? Students, MMC, all of you remember, MMC will undergo meiosis. Right? MMC undergoes meiosis to produce the microspores, to produce the microspore tetrad. microspore tetrad, which is present like this. Okay, so MMC is diploid right now. Microspores tetrad is haploid right now. So it is going to be diploid. Protonema cells of the moss is going to be haploid in nature. Okay, so if you forget that, let me just remind you, students, this is the life cycle of your mosses, and you can see, students, clearly protonema is a young stage. Protonema is a young stage present in the case of mosses, which is diploid, haploid in nature. Haploid in nature. Now this protonema forms something called, it's free, again, it is free living, green and filamentous. It is free living in nature. It doesn't have to depend on the gametophyte. Free living in nature. Okay, and this protonema forms something called as lateral buds, and these lateral buds will grow into the gametophyte eventually. So protonema is going to be haploid in nature. It's going to be haploid in nature. Okay, so who told the first answer? Who was the first person to answer in the chat? Quickly, I want to see who was the first person to answer. Students, make your mistakes again right now. Make your mistakes. Don't do it in examination. Don't do it in examination. Do all the mistakes possible. Gene, very good, Gene. Now, next question on the board. Let's see who does it first. Time starts now. Tick, tick, go. Cedrus is going to be gymnosperm. Adiantum is going to be pteridophyte. Sphagnum is going to be a moss. Markensia is going to be liverwort. So A is going to be 2. A is going to be 2. This is gone. This is gone. This is gone. A is going to be 2. D is going to be 1. B is going to be 1. Okay. C, I got the answer already, students. C is going to be 4. D is going to be 3. I got the answer already. All of you, how many got the answer, students? All of you are telling the answer already. Oh my God, all of you became smart now suddenly. All of you became more, what do you say? Like, brain cells are activated more and more now. Okay. Students, A is not the answer. Okay, A is not the answer. Okay, A is not the answer. 3 is the answer. Okay. So students, all of you keep telling me who told the first answer. Students, I can't keep a track of who's doing the first answer because I want you to keep a track of who telling the first answer. Okay. I'm the first. Harish Shreen. Oh, nice. Applause for everyone who tells the first answer now. See, biology is a game where you do the right answer in less time. So if you do less time right now, you can spend more time in your physics and chemistry. Okay. Next question on the board. Let's see who does the answer first now. Quickly, go students. Tick, tick, go. Following is incorrectly matched. Volvox, starch. Okay. Chlorophyce. Ectocarpus, fucoxanthin. Yes, makes sense. Urothrix, mannitol. No. Urothrix is going to be green algae. It is supposed to be starch shade. Mannitol is for your Phaeophyce. So 3 is the right answer here. 3 is the right answer. 3 done. Oh, nice. Who told the first answer? I think it was Yeliha Neelam. Okay, Neelam. Nice. Claps for Neelam right now. We'll applaud Neelam, students. Okay. Nice. Okay. Students, again, the whole point is to tell the right answer. If you just type random answers in the starting, inky pinky ponky, don't do that. Tell the right answer, okay? Don't do rot just because in hurry. Okay. Next question on the board. Hydrocolloid carrageen is obtained from, students. Very easy answer. I got the answer already. My answer is done, set. My answer is set already. My answer is set. Yes, clap. My answer is already set. Karthik is the first person to answer in the chat right now. I can see Rhodophyce only. Absolutely. It is going to be Rhodophyce only. Carrageen is found in the case of Rhodophyce. Rhodophyce only. Nice. Students, how long is it taking for you to solve the answers now? Because in the examination, there's a very high chance this year you will get a lot of questions from your assertion reason and as well a lot of questions from your match the following. So every question on average you should spend around 30 to 40 seconds, maximum 30 to 40 seconds in the chat in the examination. Okay, students, can I see your marks in the chat right now? Can I see your marks? Next question on the board. In which class of the algae possess pigment fucoxanthin is brown algae? Phycoerythrin is going to be red algae. Fucoxanthin is brown algae, is going to be Phaeophyce. Rhodophyce is going to be Phycoerythrin. So I got my answer already. Students. I got my answer already. Did you find your answer? Students? Tell me yes or no. Did you find your answer? Yes. Yes. Okay. Yes. Okay. Students, again, this table becomes very, very important. So, students, small game again. Every table will play a small game. The small game is very simple. I'll give you 10 seconds. I'll give you 10 seconds to see the table. After that, I'll ask you only one question and let's see who can answer. Time starts now. Go. Once you're done, just say done sir in the chat and I'll get to know, sir. I can ask you a question. Quick, quick, quick. Quick, students, quick. Quick, quick, quick. Done. 5, 4, 3, 2, 1. Time's up. Okay, students, tell me which type of flagella do you find in the case of rhodophysi? Quick, quick answer. Which type of flagella do you see in the case of rhodophysi? Which type of flagella do you find in the case of rhodophysi? Equal, apical, students, unequal. What is the answer in rhodophysi? Equal, unequal, apical, lateral. Yes, in the case of rhodophysi, absent. So don't let anyone fool you. Remember, students, this is where they ask you question examination also. They will accept, they will think you might, they'll make sure always go for such questions where they can catch hold of you. Okay, it's going to be absent in nature. Absent. Okay. Look at this question right now, students. In gymnosperms, the pollen grains are released from microsporangium and carried by air current. Yes, assertion is correct. Oh, did you see? I put 2 ticks. In air currents carry pollen grains to the mouth of the archegonia, where male gametophyte is discharged and pollen tube is not formed. Students, this reason is correct or wrong? This reason is going to be wrong. Why? Because pollen tube is formed here. Pollen tube is formed. Okay, pollen tube is formed here. So A is true, R is false. A is true, R is false right now. Okay, A is true, R is false. Done. Done, students. Option number 4. Okay, okay. Easy question, easy answer. Okay. So one is assertion reason again. The first stage of gametophyte in life cycle of moss is protonema. Remember, students, I showed you this one, right? Protonema stage, independent, haploid in nature, which forms lateral buds, give rise to a new plant. Yes, same protonema, same protonema. The protonema develops directly from spores during produce a capsule. Yes or no, students, tell me. Let me show you one more time. If you go back here and see, students, this protonema is developing from the spores. Where are spores coming from? Spores are formed inside the capsule. Inside the capsule, there is meiosis happening, and that gives spores, and these spores are developed into the protonema. Protonema develops into the male gametophyte, main gametophyte plant body. So what is the answer, students? What is the answer? Quick answer, students. Correct. Option number B. Assertion and reason are correct, and correct explanation of assertion. Okay, from capsule they're developing directly. Okay, okay. Now, students, now we come to the last 2 questions of your plant kingdom. Here we go. This is a NEET 2024 question. NEET 2024 question right here on the board. Asexual reproduction occurs by biflagellated zoospores. In the case of Phaeophyce, yes. Sexual reproduction is by oogamous only? No. We can find all 3 here. Stored food material is laminar and mannitol? Yes. Yes. And yes. So the answer is A, C, D, E. A, C, D, E. Yes. Option number 2, I guess. Option number 2. Option number 2. Okay. So option number 2 is the right answer here. A, C, D, E. Students, remember all 3 are present in the case of Phaeophyce. Oogamous only where? Oogamous only is found in the case of rhodophyce. This statement is true for rhodophyce. This is true. Okay. B4 Basavaraj Sir right now in the chat. Students, last question from your plant kingdom. Last question from plant kingdom. Here we go on the board. Last question from plant kingdom. I'm doing last kingdom from plant. Okay. Microscopic algae is going to be Ulfia. Tallest gymnosperm is going to be Sycovia. Okay. Thalloid bryophyte is going to be Markantia. Heterospory is going to be Salvia. Friends, what is the answer? Session is up to session will take a lot of time, students. What is the answer? Quick answer. A is 4, B is going to be 3. A is 4, B is 3. Only one option number 2. And students, with that, your entire plant kingdom is over. Now, students, I want everyone to tell me in the chat right now, do you feel much more confident in plant kingdom? Yes, sir. Multiple concepts got revised and I'm able to track my progress really well. So I want everyone to, I want everyone to tell me in the chat right now, students, if you are feeling a little bit confident, if you are, able to solve more questions and hopefully do not make the same mistake again. Okay. Extra marks. Okay. Students, I want to see your marks in the chat right now. I want to see your marks in the chat. Quick. What is your marks? %, sure, confidence. Session is up to, I don't know. Uma, even I don't know what time the session is up to. Yes, students. Now, I want to see your marks in the chat. All of you quickly in the chat. And I want everyone to tell me in the chat right now, plant kingdom done and dusted. Quickly, students. And I will sit for like exactly 2 minutes, exactly for 30 seconds. I'll sit. One second, students. I want everyone to type in the chat, students. Plant kingdom done and dusted. Plant kingdom done and dusted. Quickly, students. All of you. All of you, plant kingdom done and dusted right now. Because now we'll be starting. Now we'll be starting with morphology of flowering plants. Okay. Shall we start, students? Shall we start? Yes or no? Shall we start? Morphology now. Now again, students. Let me ask all of you one simple thing right now. In morphology, 90% of questions will come from where? From examples. 90% will come from examples only, and you need to find a trick for every single example to make it as efficient as possible. And let me tell you again, plant kingdom was easy, it was more conceptual. But from now onwards, you will realize that importance of memory right now in the chapter morphology of flowering plants, because every single question is memory based here. Okay, shall we start, students? Shall we start? Examples. Let's start now, students. Let's start. Examples coming. Students, I hope you remember all the examples. Okay, I hope all of you remember the examples here and there. If you don't remember, today is the day you have to finish it. Today is the day we have to remember all the examples. Okay, shall we start, students? Shall we start? All of you in the chat. Students, long back I made a video on all the examples of morphology in one video. How many of you watched the lecture? It is not there on YouTube anymore. Okay, I removed that video from YouTube. Okay, so how many of you watched the lecture where I've done all morphology examples? Every single example I've given a trick. Every single example I've given a trick there. How many of you watched that? If you've not watched it, okay, if you want that video today, students, if you want that video today, let me tell. I will upload that video. I will upload that video in the Telegram today. If you want, I will upload that video in Telegram today. Basavaraj Sir Biology. In this Telegram today, I will upload that video. I will send a link. I will send one link of that video. Okay. It is a private video. No one can access it anymore. It is not available anywhere on YouTube right now. I removed it long back. Okay. So if you want that video, I will share it in Telegram today. So join the Telegram today. I'll send the video. Link I'll send. It is a private video. Only students with the link can watch that video. Okay. So I'll send that video. Exactly. Shorts are still there. Exactly. Shorts are still there. Okay. First question on the board. Let's go. First question on the board. First question on the board. It's been removed from YouTube long back. Okay. There was some problem with it. Okay. First question on the board. Basal placentation. Basal. Birds migrate south. The trick is birds migrate south. Marigold and sunflower. So B is going to be 4. Axial. Allow China to leave. Allow China to leave. China rose, tomato, and lemon. Lemon. Okay. Birds migrate south. Sunflower and marigold. Marigold biscuit. Okay. Parallel placentation. Parents are masters. RG Mon. Right. Parents are masters. Mustard and Archimon. Free Central. Fail to change your DP. Dianthus and Primrose. Dianthus and Primrose. Tell me the answer, students. Tell me the answer. Tell me the answer, students. Tell me the answer. Option number 3. Exactly. So basal placentation is going to be what? Birds migrate south. Sunflower and marigold. Axial placentation. Allow China to leave. China rose. Parents are masters, Argimon and mustard, and free central is going to be Dianthus and Primrose. Okay. Now next question on the board. Which of the following shows world phyllotaxy? World phyllotaxy. It means, what is the meaning of world? World meaning is that, students, in one node, at one node we can find multiple leaf in one node. More than 2 leaves on one node is called as world phyllotaxy, and it is observed in the case of a stone. Which stone? Allistonia. In the case of Allistonia, we can observe that. In the case of Allistonia, we can observe that, students. We can observe that. Okay, Allistonia. Done? Done. So C for cat or C for capto? We'll think C for capto for now. Okay, C for capto in the chat, all of you. C for capto in the chat, all of you. Now, next question on the board. Okay, students, you have to remember other examples also. Here you are given, see, alternate, single leaf, China rose, mustard and sunflower. China rose, mustard and sunflower is for alternate. Calotropis and guava is going to be for your opposite. Allistonia is going to be for world. So remember these examples also, because question can come from here. Okay. C for cash also. Yes, C for cash. Okay. Now, identify the correct features about mango and coconut. Okay. In both fruit is called as a drupe, correct? Endocarp is edible, students. Okay. See, if you've eaten mango, you know, right? When you're eating mango in the childhood, what used to happen? I used to be very hungry, very hungry. So even after eating the fleshy part of the mango, I used to try eating even the inner part. It's called as the stony endocarp, right? Endocarp will be so stony, it's so difficult. Still, I used to bite the hell out of it. That's stony endocarp. Okay, but you can't eat it, but you can't eat it obviously, but it's very difficult. Very stony it is. Okay, endocarp is edible. Endocarp is not edible. You can't eat the endocarp here. Okay. Mesocarp in coconut is fibrous. Yes, coconut is fleshy. Yes. Both the fruit develop from monocarpillary ovary. Monocarpillary. Students, can anyone remind me? Is it going to be monocarpillary superior or monocarpillary inferior? All of you in the chat. Is it going to be monocarpillary superior or monocarpillary inferior? Quick. Monocarpillary superior or monocarpillary inferior ovary? Friends, quick answer in the chat. Superior ovary, exactly. Monocarpillary superior ovary. So A is the answer. You can see here, monocarpillary superior ovary. Mesocarp is fibrous. Okay. Hard stony endocarp. Okay. Next question on the board. Correct position of floral parts over thalamus in mustard is. See, first of all, students, understand. Position of floral parts over thalamus in mustard. First of all, you should tell me, students, is mustard is an example of, right, epigynous, perigynous or hypogynous? Tell me that in the chat. First of all, tell me that in the chat. Epigynous, perigynous or hypogynous? Quick. That much you tell me first of all. Epi, hypo or peri? You'll tell me, sir, hypogynous condition. Now, what's the trick for hypogynous? Hypogynous trick is hero must have brinjal. Hero must have brinjal. H for hypogynous, M for mustard, H for hibiscus or China rose, B for brinjal. Hero must have brinjal. Okay. These are the small tricks which you have to do it. So gynoecium occupies the highest position here, while other parts are situated below it. Make sense? Makes sense. Margin of the thalamus grow upwards? No, not hypogynous. Gynoecium is present in the center, other parts cover partially? No. Gynoecium is situated in the center and other parts floral rim, that is for perigynous. This is the answer here. A is the answer. All of you understood, students? All of you understood how you can approach? Yes. Understood? So you should know what they're asking you. Okay. Now let me change the question, student. Let me change the question. What if I make it to? Okay, what if I make it to? Ray floret of sunflower. Tell me the answer now. If I change the question to ray florets of sunflower or cucumber or guava, then what is the answer? Tell me, students. What is the answer then? What is the answer then? What is the answer then? Then tell me. Ray florets of sunflower. Is the answer same again or will the answer differ? The answer will change now. Answer will change right now, students. In such case, the answer is going to be, students, epigynous condition, option number 2. Option number 2. Okay. Now I'll change the question to rose. Now tell me the answer. Plum, peaches and roses. People pluck pink roses. Right. Plum, peaches or roses. Now tell me what is the answer. Now tell me what is the answer now, students. If I change the question to roses. Now tell me what is the answer. Yes. Option number 4 is the answer that time. D or option number 4 is the answer, not 3. D is going to be the answer that time. So all of you are getting an idea right now. All of you are able to answer. Tell me yes or no in the chat. All of you are able to answer. Yes or no? Quick, students. All of you are able to answer? Yes. This is how the confidence which you need to have in morphology. If you don't have this confidence, yes, I can solve any question, you'll have problems later on. So remember all the examples. Okay. Now, next question on the board. This is again is very easy. Ovary is half inferior. Ovary is half inferior. People pluck pink roses. Half inferior, students. If you don't know what is half inferior, here, perigynous condition, half inferior ovary, half inferior, perigynous. Now, students, how do I remember this? There's a small trick from my side, might not be related to you, but I remember this way. There was a cartoon called as cartoon called as Phineas and Ferb. How many of you know? There was a cartoon called as Phineas and Ferb. Okay. In Phineas and Ferb, there was a platypus. The platypus name was Perry the platypus. Okay, Perry the platypus. Perry the platypus was, if it doesn't wear a hat, it is normal platypus. If he wears a hat, it becomes detective Perry the platypus. Okay, so that platypus was half and half. The platypus was half and half. So here, every single time I read Perry the perigynous condition, in my mind, it is always Perry the platypus in my mind. I don't know why. It always happens to me. So in my mind, it is always Perry the platypus, and half the time it is a normal platypus, half the time it is a detective. So that's how I remember half inferior ovary, half inferior. That's how I remember. I don't know if you can remember in a different way. You can remember. Okay. Perry the platypus. Okay. So it's going to be plum, peaches and roses. Next question on the board, student. Next question on the board. Yes. Next question on the board. In roots that originate from the base of the stem. Let's take, this is a stem right here. This is the lowermost node here. From the lowermost node, we can see the roots arising here. From the lowermost node. In a bunch, they are found in the case of your monocots. Tell me what is the answer? Tell me what is the answer, students? That is going to be fibrous root. That is going to be what, students? Fibrous root is going to be the answer. Students, don't you feel morphology is so easy? Tell me yes or no in the chat. Morphology is actually so easy, morphology. If you know the answer, if you know the options, if you know the examples, right, there cannot be much more easier chapter than morphology. If you know the examples, it's such a laddu chapter. Nothing is in the chapter. Okay. Now, students, all of you marks in the chat before we go ahead. All of you quickly marks in the chat before we go ahead, students. Before we go ahead. Again, this question also very easy question. Very, very easy question. You see, today only I taught you what is ray florets. Sunflower ray florets have what? Okay. Inferior or half inferior, superior, super superior. What is the answer? Or under superior, whatever you call it. Hypogynous condition. Tell me. Option number D. Inferior ovary. Obviously, it's going to be inferior. All of you know guava, cucumber, and reflux sunflower. Look at this question. Which of the following is not correct? They're asking you which is the following is not correct again here. Not correct. The rhizome is a condensed form of stem. Okay. Apical bud in rhizome always remain above the ground. Okay, apical bud is above the ground. In rhizome is aerial. No, a rhizome is underground with no distinct nodes and internodes. Yes, no. There is nodes and internodes. If you drink tea, students, if you drink something called a ginger tea, and when you're trying to cut a ginger, you might notice a ginger has all these lines on them. A small lines. There are small lines present on the ginger. There are small lines present on the ginger, and these lines are nothing but no internodes here, nodal regions here. So in ginger we can find nodes and internodes can be found. Okay, so C for capto here, C for cash. Okay, next question again, students. Again, question based on example. Imbricate, advocate. Advocate fights gun cases. Gulmohar and Cassia. Gulmohar and Cassia. Valvate chair, Calotropis. Okay, Vexillary estivation, bean. Okay, BP vaccine, bean and pea. Twisted, lady from China has a twisted cotton. Lady from China, I'll write the trick here, students, if you don't forget. Twisted is going to be lady from China has a twisted cotton. Lady is lady's finger. China rose. Twisted estivation. Cotton. So cotton, China rose, lady's finger are the examples of your twisted estivation. Twisted estivation. Okay. Everything is given here. Everything is given here, students. Which of the following plants possess placentation of ovules born on central axis with no septa? Free central. Which of the following is free central basically? They're asking you for free central here. Free central right here. Quick. Lemon. Students, do you know why I say lemon? Because in my class there was a guy called Aman. Okay, Aman. He is right now a teacher in one of these top offline institutes. Okay, and when he came to college for the first day, and all the boys we are like, we'll go drink lemon tea, and he used to call it lemon tea. So throughout the 2 years, he made fun of him for calling once lemon as lemon. Okay, till now the habit has not gone for me also, because that's how we used to tease him. Lemon tea. So I hope he's not watching lecture. I hope he never sees his lecture, because I'm still making fun of him. Lemon tea. I still remember this was in MSc first year, first day. He by mistake he told lemon tea as lemon tea, and till now we still catch hold of him for that. I don't know. It always happens in boys. We catch hold of someone. If someone says something once wrong, throughout the year it's going to be stuck. Throughout 2 years it's stuck with them. Lemon tea. Now all of you also know why I call it lemon, because of that person. Okay. Okay. Which of the following plant possesses placentation of ovules born on septa? Free central. Fredo, change your DP. Fredo, change your DP. Okay. Exactly. Gingerly oil is for me. Okay. Fredo, change your DP. Dianthus and primrose. Dianthus and primrose is the example for free central. Free central. Free central placentation. Okay. Free central placentation is going to be primrose. Easy students, easy. Oh god. Okay, choose the mismatched pair of leaf characters with its example. Mismatched pair. Okay. Permanently compound allostonia. There only one scam is there. First question only. Okay. Alternate phyllotaxy, China rose, leaf tendrils, pea plant, opposite phyllotaxy, calotropis. Students, find the scam first. Find the scam first. First option only scam is there. All of you know allostonia, right? Allostonia has world phyllotaxy. Allostonia has world phyllotaxy right now. Right. World phyllotaxy. So A for Ames, A for Amumam, A for ant. I don't know what is A for aeroplane. Okay. A is the answer. Okay, again, this is a very good question. Let's see who does it first. Okay. Competition starts, time starts now. Who does it first? Identify the correct statements related to androecium. Related to androecium in a flower. Stamen, sterile stamen is staminode, yes, correct. When stamens attached in petal, they are called as epipetalous condition, yes. Monoadelphous is seen in the case of China rose, yes. Polyadelphous in the case of pea plant, no. Polyadelphous is citrus, pea plant is going to be diadelphous condition. Variations of length of anther filaments is seen in the case of mustard. Mustard, students, A, B, C, E, A, B, C, E, A, B, C, E. A, B, C, E, students, A, B, C, E. A for apple. Oh, A for apple. Students, I forgot A for apple. Forgot. A for apple is the answer. Now here B for what, students, do you remember what was B for? B for bat. If I don't remember, if I remember correctly, B for bat it was. I don't even remember what was B for actually at this point. Okay. So all of you understood the difference, right? Here this is going to be wrong here because polyadelphous is seen in the case of where? In the case of citrus. In the case of citrus. Pea plant is diadelphous. Pea plant is diadelphous in nature. Okay. Remember students, in the case of your epiphyllous condition, lily is the example. Remember that also. Lily in the case of epiphyllous condition. What is epiphyllous condition? Epiphyllous condition is a condition where the stamens are attached to the perianth. Attached to the perianth. That is called as epiphyllous condition. Okay. Students, smash the like button, all of you. What is this? Again, very good question. Let's see who does it first. Time starts now. If you're answering after me, like the video otherwise. If you're answering before me, you can keep quiet. Okay. Leaflets of modified into pointed thorns in citrus and bougainvillea are wrong. Okay. Axillary bud forms slender and spiral coiled tendrils in cucumber and pumpkin. Yes. Stem is flat and fleshy in the case of your functions of leaf. Yes, correct. Rhizophora shows vertical growth. Yes. For the respiration. Yes. Subaerial growth stem in grasses and strawberry helps in vegetative propagation. Absolutely correct. Students, I got my answer. I got my answer, students. Before ball, yes, exactly. I got my answer, students. I got my answer. I got my answer, students, already. I got my answer. B, C, D, E. For me, option number 4 is the answer. What is your answer? Should I explain or should I not explain? Tell me, students. Should I explain or not? Listen now, carefully. This is going to be wrong. Why? Because it is not leaf modifications are pointed to thorn. No. It is supposed to be stem modification here. Stem modification. So this statement is wrong here. Wrong statement. Axillary bud forms that, yes, axillary bud develops into cucumber, pumpkin, grapevine and everything. Grapevine and everything is going to be there. Stem is flat and fleshy in the case of your opentia and as well as your what is that? Flat is opentia. Cylindrical is euphorbia. Cylindrical is euphorbia. Remember, cylindrical is euphorbia. Flat is in the case of opentia. Rhizophora shows vertical growth. Yes, it shows vertical growth, obviously. Okay. Subaerial growing stems like grass and strawberry. They will show something called a stolon. Stolon, as well as your what is that term? Runner type of your vegetative propagation can be seen there. Very easy, students. So the first statement is going to be wrong because it's supposed to be stem here, not leaf. That's a small difference which you need to know. Okay. Okay. Friends, I hope it is visible to all of you. It is not visible to all of you. I'll read it out loud. In pea plant, there are 5 petals. There are 5 petals in pea plant. Okay. Pea plant. 1 plus 2 plus 2 arrangement can be seen here. The question is asking you what is each plant called as? Or what is each petal called as? This one is what? This is what? This is what? Time starts now. Time starts now, students. What is what they're asking you basically? What is what they're asking you right now? 1 plus 2 plus 2 arrangement. What is the outermost petal called as posterior one? Then 2 lateral and 2 interior. What are they called as? What are they called as, students? Standard wing and keel. Standard wing and keel. Yes, option number 4. Standard wing and keel. Okay. In calotropis estivation, students, calotropis, velvet chair, velvet C for calotropis, velvet calotropis. Students, what is your marks in the chat? Can I know your marks in the chat, students? This is very student. At this point, I'm feeling lazy to solve these questions. I don't know why. I am feeling lazy to solve these questions right now because all the questions are so repetitive year after year. And let me tell you, these questions are not random questions. All of these questions are NEET PYQs. So the questions are so repetitive now. At this point, every question is so repetitive. You can easily tell what is the what answer. It doesn't even take effort at this point. Tell me yes or no. Friends, tell me all of you agree with me. All of you agree with me, students. There's no effort required to solve morphology questions anymore because morphology is so easy if you know the examples. It is so easy. It's so easy. Okay. This is the question you see. A flower is a modified shoot, yes, where shoot apical meristem becomes floral meristem, correct here. Okay. Internode of the shoot gets condensed, internode is getting condensed to produce a different floral appendage laterally as successive nodes instead of leaves. So here at the nodes we have floral appendages, we do not find leaves. Yes. Option number 2. Option number 2. Okay. Now this is a direct line of your NCRT. Can you see a direct line of your NCRT? Flower is a modified shoot where shoot apical meristem changes to floral meristem. Internodes do not elongate and axis gets condensed here. Okay. Axis gets condensed here. Done. Next question on the board, friends, quickly. Next question on the board. Axial placentation is seen in. Allow China to leave. Allow axial China rose, lemon and tomato. Instead of tomato, there is petunia given here. Instead of tomato, petunia is given here. Instead of tomato, petunia is given right here. So 1 is the answer. 1 is the answer. Students, tell me the answers. All of you sleeping now or what? All of you sleeping now? Huh? Tell me, all of you tell me the answer now. All of you tell me the answer. Yes? Everyone? Everyone tell me the answer. And all of you quickly smash the like button, all of you. All of you quickly smash the like button first of all. Done? Done. See this question. Identify the, this is a NEET 2024 question. NEET 2024 question right here on the board. Okay. Identify the type of flower based on the position. Can you see, students? In both the cases, ovary is present in the center. And all the floral appendages are arising from the rim of the thalamus. Both are going to be what? Both are going to be perigynous in condition. Half inferior condition. Half inferior condition. Perigynous. Again, NEET 2024 question. Which of the following is an example of actinomorphic? MCD is for actinomorphic. Mustard, datura, chili. Mustard, datura and chili. Datura is the answer. Datura is the answer, students. Datura is the answer right now. MCD is the for actinomorphic. For your zygomorphic, it's going to be BGP. Candidate from BGP. Our new party is here. BGP party, not BGP party. Okay. Candidate is going to be your cassia, bean, gulmohar. Right. P, bean, gulmohar and cassia. P, bean, gulmohar and cassia. P, bean, gulmohar and cassia, can the one be here? Okay. Datura is the answer. Now, answer in the chat quickly, students. Answer in the chat. Identify the part from the seed given below, given figure which is destined to form root. Obviously, students, this is radical here. Radical will develop into root. Develop into root. Student, what happened to the pen here? Suddenly, arrow mark is coming. And here, if you notice, students, D is for your coleorhiza. C is for radical actually. C is for radical. Okay. So, as I said, D is for radical. This is what confusion happened in exam also, I feel. Student, this was a confusion here. This is supposed to be, C is supposed to be for radical here. This entire thing is radical. C is for radical, sorry. D is for, correct only I told, no. C is for radical. D is for coleorhiza. D is for coleorhiza. Yes. D is for coleorhiza. Correct, correct. Student, let me just check the question. Match the following, students, again match the following. Monoadelphous in nature is going to be your China rose. Diadelphous is going to be pea plant. Polyadelphous is going to be citrus. Epiphyllus is going to be lily. Epiphyllus is going to be lily. A is going to be 4. B is going to be 2. C is going to be 1. And D is going to be 3. Again, do you realize same type of question has come every single year? See, again, match the following. See, this type of questions you can expect in this year also. These type of questions are very important because this will check your example remembering capacity for every single question. See, rose. Okay. People pluck pink roses, perigynous condition. A is going to be 2. Pea plant is going to be marginal placentation. Cotton is going to be twisted estivation. Mango is going to be a droop. Yes or no? Let me yes or no, students, in the chat quickly. Rose is going to be perigynous condition. Pea is going to be your marginal placentation. Cotton is going to be twisted. Mango is going to be droop here. Mango is going to be droop here. Answer is 4. Answer is 4, students. Look at this question again, student. This is a NEET 2024 question. This is again a NEET, re-NEET or something. I probably re-NEET. Question on the board. In cymose inflorescence, the flowers are born in acropetal position. No. First statement only is wrong here. Why? In acropetal is for racemose. Acropetal is for racemose here. Okay. Acropetal is racemose. Okay. Gulmohar flowers are actinomorphic. MCD. Students, quickly, students. Gulmohar is an example of actinomorphic. Actinomorphic is MCD. In flowers of cucumber, margins of thalamus grow upwards, enclosing, completely getting fused with other parts of the flower. Yes, epigynous condition. Guava is an example. Bracts are reduced leaves to form the base of pedicel. Yes. Flowers are hypogynous in mustard. Yes. CDE. 3 are answer. This is wrong because, students, Gulmohar is an example of zygomorphic. Gulmohar is an example of zygomorphic. Actinomorphic is what is the example, students? Actinomorphic is MCD. McDonald's. Mustard, Datura chili is examples of actinomorphic. Mustard, Datura chili is an example of actinomorphic in nature. Okay. Actinomorphic is MCD. Now, negatively geotropic roots. Very easy question, students. Very easy question. New metaphors. Very easy question. New metaphors. Doesn't even require. NEET 2024 question. Easy question also comes like this from sometimes. Okay. Last question, students. Last question from morphology. Last question from morphology on the board. Polyadelphous condition. More than 2 bundles. Okay. Syncarpous condition, carpels are fused. Staminode, sterile. Epipetal condition, attached to petal. So A is going to be 1, B is going to be 4, C is going to be 2, D is going to be 3. Answer number 1. Answer number 1. And students, with that, morphology is also over. With that, your entire morphology is also over. Students, how do you feel about morphology now, students? Tell me in the chat. How do you feel about morphology? Are you feeling confident in morphology? Tell me yes or no in the chat quickly, students. Are you feeling confident in morphology now, students? Yes, morphology is difficult for all the students who do not know the examples. If you know the examples, if you know the diagrams in morphology, morphology is the easiest chapter to score marks. So all of you students in the chat, hashtag morphology done and dusted now. Hashtag morphology done and dusted. All of you in the chat. Hashtag morphology done. Yes. Hashtag MOFP done. Hashtag morphology of flowering plants done. Hashtag MOFP done now. Done, students? Done? Morphology done now. Now, students, can we start anatomy? After anatomy, I'll give you a small break because my leg is also hurting a little now already. My leg is also hurting a little, so I'll give you a small break after anatomy. Anatomy is very small, and anatomy PYQs are a little less here because in anatomy, majority of the questions used to come from secondary growth. Secondary growth is removed right now. Secondary growth is removed, so questions are a little easy only in anatomy right now. Anatomy used to be difficult. Once upon a time, anatomy used to be difficult. If you think about secondary growth, since secondary growth is removed, I don't know why. So it's very easy now. Okay. Now, first question on the board. Regeneration of damaged growing grass following grazing is largely due to regeneration of damaged growing grass. Okay, is largely due to intercalary meristem, apical meristem, lateral meristem, secondary meristem. It's going to be intercalary meristem, students. It's going to be, students, intercalary meristem. Intercalary meristem, especially in the case of grasses. In the case of grasses, see here, students. The meristem which is found between mature tissue is known as intercalary meristem. They occur in grasses and regenerate parts removed by the grazing of herbivores. So when the herbivores are, when the cow is coming eating nicely, nom nom nom, it is eating. It will again grow back because of intercalary meristem. Because of intercalary meristem. Next question on the board. Phloem in gymnosperm slacks. In gymnosperm, right? Phloem in gymnosperm lacks. What is missing in phloem basically? What is missing in phloem? CO tubes and companion cells. Yes, CO tubes and companion cells both are missing. Both are missing. Again, very easy question, students. Again, very easy question. Both CO tubes and companion cells both are missing in the case of phloem. So here in phloem, apart from companion cells and CO tubes, they have something called as albuminous cells and sieve cells are present in the case of gymnosperms. In the case of gymnosperms, they are present. Okay. See, gymnosperms have albuminous cells and sieve cells. Right? They lack sieve tubes and companion cells. Both are absent here. Both are absent here. Gymnosperms have albuminous as well as sieve cells. These both are present here. These both are absent here. Okay. Next question on the board. The grasses. All of you might have seen, students, in whenever there is no water, whenever there is no, let's say there is no water at all, water is missing. If water is missing, what happens is all the grasses, right, the leaves will start to curl inwards. This way, the leaf will start to curl inwards. Why? Because there is a large empty colorless cell. The large empty colorless cell will lose all the water. The large empty colorless cell will lose all the water, and that cell is called as what? Bulliform cells. Bulliform cells will lose all the water and the leaf will start to curl inwards. Curl inwards. Okay? Curl inwards. Bulliform cells. Adaxial epidermal cells along with veins modify themselves to form large empty colorless cells called as bulliform cells. Okay? A transverse section of plant shows the following anatomical features. Student, the first thing you should tell me what is it. Okay. large number of scattered vascular bundles surrounded by bundle sheet. Scattered vascular bundles here. Scattered vascular bundles. Scattered vascular bundles here. This is a typical example of your, who can tell me students? Typical example of your monocot stem. It's going to be your monocot stem. See students, this is a clue here for monocot stem. This is the major clue for monocot stem. That is, phloem parenchyma is absent. Without even looking at the options, you can tell me, sir, it is monocot stem. It is going to be monocot stem. If this statement is written here, okay, this statement is written here. Again, you can tell it is a monocot because it is closed. If it is closed, it is monocot because cambium is absent here. Okay, cambium is absent here. Okay. The type of tissue commonly found in fruit walls of nuts. A type of tissue which is found mainly in the fruit walls of nuts. Then 50% I'll tell you why it is called 50% here. Let me tell you why it's 50% here. 50% students will go for sclerenchyma. 50% will go for sclerets. But what is the answer? If you look at your NCERT lines, it says right here, right? The sclerets are spherical. Sclerets are type of sclerenchyma, oval, cylindrical and highly thickened dead walls with very narrow cavities. These are commonly found in fruit walls of nuts, pulp of fruits like guava, pear and sapota, seeds, coats of legume and leaves of tea. So this is very, very important if you have to specifically tell the answer. Sclerets is the answer there. The answer. Okay. Again, a very easy question, students. As soon as you see initiation of lateral roots, directly in your mind you should get one word: pericycle. Initiation of lateral roots and vascular cambium during secondary growth is take place in the case of your pericycle. In the case of pericycle, option number D. Students, can I get to know your marks in the chat? Karthik, Koplu, Swapna, Saura, yes, Ravindrani, Meghana, Amy, Usha, what is your marks in the chat? What's your marks? Can I know the marks in the chat, all of you? Students, first of all, please smash the like button. Can I know the marks? All of you? Drink some water. Students, do you realize then we have done almost 50 plus questions already? Do you realize we have solved more than 50, 55, 50, 60, almost approximately around, let's say around 60 questions approximately? Yes? Emperor? At least count now. Okay. Students, do you want a part 2 for this video? Before we go ahead, do you want a part 2 where we do all botany from your class 12th, because class 12 botany is very important, especially chapters like your, I think ma'am will do 60% flowering plants, like chapters like your genetics and microbes, entire ecology, principles of inheritance, human reproduction. Do you want? Same way, we will make you realize, we will check how much you have known. See, this type of sessions will give you a insight on how much you're able to answer, in what areas you need to improve. For example, if you're not able to solve questions from a particular chapter, you can go back and start doing that chapter, exclusively that chapter. Okay, that is the advantage of solving PYQs. Okay, that is the advantage of solving PYQs. Okay, next question on the board, student. Next question on the board. Which the following is not a character of collenchyma? Students. Collenchyma, which is found in the case of your petiole and young stems. Okay. Now, before we go, students, can I ask you all of you a simple question? Do you find collenchyma in the case of monocots or in the case of roots? Do you find collenchyma in the case of monocots as well as roots? Quick, students, quick. Do you find collenchyma? No, you do not find. Okay. Mechanical support, below epidermis, thick corners. Wrong, because They are not dead, they are living like us. They're going to be living like us. They're found only in the case of dicot, that too only stem and root. That too only stem and root, nowhere else. Nowhere else. Okay, very exclusive. Premium subscription it is, very premium it is. Okay. Axillary buds are derived from the activity of, for example, there is something growing this way. As it is growing up and up and up, some cells are left behind. As it is growing up, some cells are left behind like this here. Some cells are left behind here. Now these cells eventually develop the axillary bud. They form the axillary bud eventually. So what is it, students? What is the top part is growing on top? That is called as your apical meristem. So as the apical meristem is growing, some cells are left behind. That is called as axillary bud. That makes up the axillary bud, students. Axillary bud. Beautiful, beautiful. Done. Easy question. Looks big, but very easy question. Let's break it down one more time. All of you students, solve this question with me. I want everyone to solve this question with me now. Okay. In roots, xylem and phloem in vascular bundles are arranged in alternate manner. Yes, in alternate xylem, phloem, alternate manner in different radii. Yes, it is called as radial arrangement in root. Correct. Conjoined closed vascular bundle do not possess cambium. Yes, if it is closed, no cambium. Makes sense. Makes sense. In open vascular bundles, cambium is present. Yes, open means cambium is present. The vascular bundles of dicot stem possess endarch. Yes, see, stem is always endarch. Yes, root is exarch. Stem is endarch. Okay. In monocotyledonous root, usually there are more than, see, monocot, we can see polyarch condition. We can see polyarch condition in the case of monocot. So correct here. So what is the answer, students? What is the answer? What is the answer, students? Option number D. All statements are correct. So all of you should be able to follow what I am saying. If you are not able to follow any of the students, please watch my lecture, Anatomy of Flowering Plants in 3D. How many of you watched it, students? How many of you watched it? How many of you watched my lecture, Anatomy of Flowering Plants in 3D? That is the best lecture out there for anatomy. Let me tell you, that's my confidence, because the way I have approached the chapter, the way we have taught you the entire anatomy there, right? It's really amazing job there. So please watch it once. Okay, Anatomy of Flowering Plants in 3D, just watch it once. You will think anatomy is the easiest. You will think the anatomy is easiest there. Next question on the board, students. NEET 2023 question. Okay. Consider the following plant tissues. Okay. Following plant tissues are here. Okay. Axillary bud, fascicular vascular cambium, interfascicular cambium, cork cambium, intercalary meristem. How many of them are lateral meristem? The question is, how many of them are lateral meristem right here? Lateral meristem. Axillary bud, no. Okay. No. BCE. Intercalary meristem will be no. Now makes sense. For a second I was like, what is that? Students, lateral meristem will have cambium in it. Okay. So fascicular vascular cambium, interfascicular cambium and cork cambium. All three are examples of lateral meristem. Watched it many times. Huh? Oh God. Okay. The transverse section of plant shows polyarch condition. Students, from this polyarch, what you can tell me, students, what is the answer? Radial exarch. Exarch means root. Monocot, dicot root. Polyarch means monocot root. Monocot. Again, radial also you can tell it is going to be root only. Okay. So it's going to be a monocot root. Monocot root. You can easily tell monocot root. As soon as you see polyarch condition, you can tell me it is a root. Without even thinking, you can tell me, sir, it is going to be a root only. Okay, it's going to be root only. Okay. Now, endarch and exarch are terms often used to describe the position of secondary xylem in the plant body. Exarch is common feature in the case of root. Yes, exarch in the case of root, endark in the case of stem. Okay, so tell me the answer, student. Tell me the answer. Yabba. Students, what is the answer? I'll tell you what is the scam here, students. I'll tell you what you need to look at here. This is the scam here. Why? Because secondary xylem does not have that all this configuration. It is only found in the case of primary xylem. It is found only, it should be primary xylem, not secondary xylem. This exarch, endark, all present in primary xylem only. Secondary xylem, part of secondary growth, does not have any of this. Does not have any of this. Okay. So this statement becomes wrong here. This statement becomes wrong. Okay. So what is the answer? A is incorrect, B is correct. A is going to be incorrect, but B is going to be correct here. So this is the scam here. Okay. Look at this question, students. In the given figure, which component has thin outer wall and thick highly thickened inner walls? Obviously, it's going to be your guard cells. Guard cells outer is thinner, inside is thicker. Why inside is thicker, students? Because inside is thicker because it needs to hold the pressure of all the water, water vapor coming out. So answer is going to be option number 4 and it's going to be C. Option number 4, C is the answer. Okay. Option number 4, C is the answer. See, D cannot be the answer because D is the pore. Pore cannot have that. D is the pore. Pore cannot have it. Here, C is pointing the guard cells. C is pointing the guard cells here. Guard cells will have that. Okay. Guard cells will have the entire configuration. Okay. Parenchyma is living, but collenchyma is dead. Again, wrong, students. Collenchyma is living. How many times to tell you? I don't know. Gymnosperms lack xylem vessels, but the presence of xylem vessels is a characteristic of angiosperms. Yes. True. True. Okay. Gymnosperms will lack xylem vessels. They like the xylem vessels. So one is false, true is correct. Okay. Boliform cells are responsible for inward curling of leaves in monocot. Inward curling leaves in monocot. Blunt salt stress. No. Increase. Nothing. So option number 4 is the right answer. This is a NEET 2024 question. NEET 2024 question. Very easy question again. Very easy question. Very easy question, students. Okay. Students, I hope it is visible. If it is not visible, you are supposed to tell the order here. You are supposed to tell the order. You have to make up the order. I'll give you 10 seconds. I'll give you 10 seconds. Make up the order. Tell me the answer. Make up the order and tell me the answer. The question is starting from periphery. Starting from periphery to inwards, you should go. From outside, from here, from the periphery, you should go inwards. Okay. Periphery to inwards. Tell me the answer. Periphery to inwards, students. Periphery to inwards. See, outermost is going to be what? Epidermis. Then what do we have? Cortex. Then what do we have? Endodermis. Then what do we have? Pericycle. Innermost is going to be pith. See, innermost has to be pith. So these two options are gone automatically. Okay. Here from pericycle, do we say from pericycle? No, not pericycle. So easy answer. Easy answer. Easy answer it becomes. Okay. Now, given students who told the wrong answer, students don't tell the wrong answer so easily. Sheila, understand. obviously the innermost, the inside one is going to be pith only. So B has to be the end, right? Right. Innermost is going to be pith. So this cannot be the answer. This cannot be the answer. Both of these cannot be the answer right now because pith has to be in the last. So B. Okay. Then what do we do? Where do we start? We start from outermost. That is epidermis is the outermost. So C is starting. Students, answer visible or mistake? Answer visible or mistake, students? Mistake, answer is visible. It's a vessel. It's a vessel, students, vessel. See here, NCERT is giving you, see, phloem is not given you a syllabus, only vessel is given you a syllabus. Tracheids is given here, tracheids and vessels. Tracheids and vessels is given. How do you remember, students? How can you remember? Tracheid is going to be a small TT. Tracheid is going to be a small tap in your house. Vessel is going to be a big drainage pipe. Okay. Look at this question. Companion cells help in maintain the pressure gradient in CO tubes. Yes. Gymnosperms lack vessels. Yes. Xylem vessels are devoid of cytoplasm. Okay. Septate aseptate. Yes, it can be septate aseptate. Mature sieve elements in phloem possess cytoplasm. Oh ho ho. Last option E is, E looks very suspicious, students. E looks very suspicious. E looks very suspicious, students. E looks very suspicious. See, companion cells help in maintaining the pressure gradient in CO tubes. Yes. In CO tubes, you can see this diagram only here. You can see, students, companion cells are present along with CO tubes here. Sieve elements it is present next to it. They got sister elements. So this becomes correct here. This becomes correct here. So if A is going to be correct here, okay, this option is gone. This option is gone. So these two options are gone here. These options are gone right now. Now you see. Okay. Gymnosperms lack. Yes. This is correct. This is also correct. Devoid of cytoplasm. This is also correct. This is wrong. Why, students? Because mature sieve elements will not have phloem. They will not have phloem. They'll become like a normal tube. They'll become. Everything is transported there. Everything is transported there. Okay. So this is going to be in phloem. Cytoplasm vacuole. No. Doesn't have nucleus. Doesn't have anything. It's a normal pipe there. So E is present. E is. In colon-chyma, cell walls are thickened at corners due to deposition of cell wall, hemicellulose and pectin. Yes. Sclerenchyma consists of lignified cell wall and pulse pits. Yes. Again easy, students. Again easy. Again it's very easy. Students, with that, your entire first 4 chapters are over. In approximately, let's say, 1 hour 45 minutes, first 4 chapters are over. Now we'll take a small 15 minutes break and we'll come back. Okay. Not 15, let's just take less than 15. We'll start class 12. Class resume at 12. Okay. Okay. We'll resume at 12 p.m. Okay, we'll resume at 12 p.m. and we will start with something called a cell unit of life, biomolecules and cell cycle. 3 chapters we will do. After again we'll take a 10 minutes break. 10 minutes break we'll take and then we finish plant physiology. And with that your entire will be over. So students, I want everyone to type in the chat right now. Anatomy done and dusted, all of you in the chat. Anatomy done and dusted. Anatomy done and dusted in the chat, all of you. Quickly students. So I will see all of you again back at 12 p.m. Do not run anywhere. Okay. Do not run anywhere, students. Come back. Let's take 15 minutes break. Okay. We'll come back and we'll start again. Okay. Let me just rest my legs a little because leg is a little hurting now. Okay. So I'll just take a 15 minutes break and come back. Okay. You know what, students? Let's make it. 1 second, students. 1 second. Make it 12:10. I'll just take 10 minutes more break. I'll just apply some cream to my leg and come back. Okay, let's make it 12:10. Okay, students. Anatomy done and dusted. We'll come back at 12:10 p.m. Okay, 12:10 p.m. We'll come back and we'll start. Okay, students. Sure, then take a break, chill for some time. So hello students, welcome back. Welcome back students. Break is over now. I want everyone to quickly go and call all your friends and ask them to come back again. And we'll be starting with a new unit altogether right now called as cell unit. Now again, students, let me tell you, this entire unit is very, very important, especially the chapters like your biomolecules, which carry a lot of weightage and the questions are a little tricky here. And especially chapters like cell cycle, very easy chapter, and you can get easy marks, free marks from the cell cycle. So with that being said, students, with that being said, can we start, students? Can we start? Can I get some fire emoji in the chat to let me know, sir? Yes, we are ready to start solving questions from the chapter Cell, the Unit of Life right now. Shall we start, students? Shall we start? Can I get a thumbs up in the chat? Thumbs up, fire emoji in the chat, or anything in the chat to let me know, yes, sir, we are ready right now. Yes. Shall we start? Okay. The first question on the board right now. Again, students, here 90% of questions will come from organelles and what are the functions of organelles. Apart from that, there will be nothing else here. Don't worry. Very easy now. Very easy. Okay. Golgi body. What is the function, students? Golgi body is formation of glycoprotein and glycolipid. Lysosomes is digesting molecule. Yes. Golgi body is formation of glycoprotein. Lysosomes is a digesting molecule. Vacuole is trapping of waste, extruded product. Ribosomes helps in synthesis of your protein. So A is going to be 3. A is going to be 3. Okay. Let me just remove the other options. This is gone. This is gone. Okay. B is going to be 4. Yes. C is going to be 2 and D is going to be 1. So option number 1 makes more sense here. Option 1 makes more sense. Let's go. Yes, let's go. First question, students, I want everyone to keep a track of your marks. Keep a track of your marks in the chat because that will show how much of marks you're gaining, how much of marks you're losing. Okay, students. So keep a track of your marks. It's very, very essential. Very essential. Okay, very essential. Now, students, next question on the board. Here we go. Which of the following nucleic acids is present in an organism having 70th ribosome? Again, students, this is also true for mitochondria and chloroplast. So here they're talking about prokaryotes. Here they're talking about your prokaryotes. Here prokaryotes, which has 70th ribosome. Prokaryotes have 70th ribosome. All of you know that. So in 70th ribosome, right? Prokaryotes. Which type of DNA do you find here? Which type of DNA do you find? So if you tell me, students, if it is double stranded, yes, double stranded. First of all, it is going to be double stranded. Okay. Not who told students? Who told single stranded? No, we don't find single stranded. Who told you single stranded? First of all, okay. It says single chromosome can be found or circular DNA. Okay, the single chromosome, but it is double stranded always. It is always double stranded. Okay, it is double stranded. Who told you single stranded? Okay, in the case of prokaryotes, we can have double stranded DNA is present, but it can be circular or straight. Okay, it can be circular or it can be linear in nature. Double stranded DNA. Okay. Okay. Next question on the board. Next question on the board, students. A non-membranous nucleoplasmic structure. Non-membranous nucleoplasmic structure in nucleus are site for active synthesis of. Let me tell you, students, they are talking about a structure which is present inside nucleus. Present inside nucleus. The structure which is present inside nucleus they're talking about here is nucleolus. They're talking about, students, nucleolus here. So can anyone tell me what is the main function of nucleolus right now? The main function of nucleolus. Anyone in the chat who can tell me? The main function of nucleolus. What is it? What is it going to be? Main function of nucleolus right here. Nucleolus. Anyone? Quick. Function of nucleolus. It's going to be rRNA synthesis. Very good. It's going to be, students, rRNA synthesis. Remember, inside nucleus there is nucleolus. Nucleolus is a structure which does not have any membrane, and the main function is production of your rRNA. Okay. Now students, you can see very clearly the content of nucleolus continues with the rest of the nucleoplasm. It is not a membrane bound structure. It is a site for rRNA, ribosomal RNA synthesis. rRNA synthesis is happening here. rRNA synthesis happening. Okay. rRNA synthesis. Larger and more numerous nucleoli are present in cells actively carrying out protein synthesis. Okay. Next question on the board. Which of the following statement is not correct here? Not correct statement here. Lysosomes are formed by process of packaging of students here only. All of you should know, students, by now. Lysosomes are formed by packaging of not endoplasmic reticulum but Golgi complex. Golgi complex helps in packaging. It's Amazon factory. They help in packaging here. They help in packaging. Okay. Okay. So option number 1 is the right answer. 1 is the right answer. Next question on the board. See here. Which of the following pair of organelles does not contain DNA? Does not contain DNA. Nuclear envelope mitochondria. Mitochondria has DNA. Mitochondria has DNA. Chloroplast has the DNA. Lysosomes and vacuoles, both of them do not have the DNA. So option number 4. So option number 4. Okay. Gopika ma'am has done plant human physiology. Check that out. Okay. So option number 4 is the right answer. It does not have DNA. See, mitochondria has DNA. Chloroplast has no DNA. But lysosomes and vacuoles do not have any type of DNA present in them. Right? Doesn't have any type of DNA present in them. Now look at this question, students. The shorter and longer arm of submetacentric chromosomes are referred to as. So there are two arms, all of you know. P arm and Q arm. Which is P arm, which is Q arm right here? The question is, which is a P arm and which is a Q arm right here? Okay. The shorter arm is P or longer arm is P? Quick. I'll give you 10 seconds. I'll give you 10 seconds to answer this question now. Just 10 seconds to answer this question now. Exactly. P arm, Q arm. Here you can see, students, in submetacentric, P arm is always shorter. P arm is supposed to be shorter here. So P arm is shorter, Q arm is longer. So P arm is shorter and Q arm you can see longer in the case of submetacentric. Now students, in the case of metacentric, what is the situation? In the case of metacentric, students, yesterday mock test questions are coming here. Your mock test questions are, I am doing mock test question paper also. Okay, questions are also coming up here. So students, in metacentric, do you think they're equal or unequal again? In the case of metacentric, they're equal. P arm and Q arm are equal in the case of metacentric. Okay. Now, the concept of omnicellular e-cellular regarding the cells divided from pre-existing cells was first proposed by who was a scientist who came later on told us that all of you are wrong. I will tell you what exactly is how the cells are coming into existence. That was nothing but Radolf Virchow. It was Radolf Virchow who told us that cells exist from pre-existing cells. Omnicellular e-cellular. Okay. Look at this question, student. Look at this question now. The size of PPLO, pluro-pneumono-like organisms, is what is the size of PPLO? What is the size of PPLO, students? Again, memory-based question. Again, a memory-based question right here. Students, I want everyone to tell the answer. I want everyone to tell the answer, regardless if you know the answer, if you don't know the answer also, you should tell the answer in the chat. Because I want everyone to make all the mistakes right now. Do not make these mistakes in examination. Do not make these mistakes in examination, so everyone has to attempt. If you know, if you don't know, that is secondary. If you know, you don't know, that's secondary, but try attempting. You are not losing any negative mark right now. The answer is option number 4. You can see PPLO is around 0.1 micrometer. You can see here 0.1 micrometer present right here, students. If you know the answer, you know it right now. If you don't know answer, you can attempt and understand and rectify again. Yes or no? You can rectify your answers. Next time if they ask you question again, see. Now students, again, attempt this question one more time now. Tell me the answer right now, students, again. Tell me the answer, all of you. Students, tell me the answer right now again. Quick answer I want. Now tell me the answer. What is the size of PPLO? You'll tell me, sir, option number 4. Why are you telling 3 again? Again, option number 4. 0.1 micrometer. No. So they can ask you for typical bacteria, 2 micrometer, viruses, eukaryotic cell. Apart from the students, they can ask you RBC also. RBC they can ask you. Okay, RBC also. Students, don't worry, a result will come as it is supposed to come. You cannot change your result right now. You cannot change your result. Result will come as such only. Okay. Now students, next question on the board. RNA synthesis occurs in. See, this is a question which is reframed right now. Reframed question right now. Right? Reframed question right now. All of you. Reframed question. The biosynthesis of RNA occurs in your nucleolus. Happens in the case of nucleolus. Very easy now. Very easy. Happens in the case of nucleolus. It becomes very, very easy for you to answer now. Very easy for you to answer. Same thing is repeating. Same story, different day. Nucleolus. Look at this question now. Inclusion bodies of blue, green and purple and green photosynthetic bacteria are. They're basically type of what? They're basically type of inclusion body. They are type of inclusion body which helps in storage. But which type of inclusion body? There are multiple types of inclusion bodies are there. Multiple types are there. Right? So this is which type of it? Who can tell me, students? Gas vacuole? Yes, absolutely. Correct answer. Gas vacuole. It's a type of gas vacuole. So, students, again, I want everyone to attempt the answer. Try attempting. Do not be like, sir, I do not know the answer. I'll simply keep quiet. No. Try attempting the answer. If you know the answer, correct. If you don't know, you learn. That's all. So, answer is option number 2, gas vacuole. Now, again, students, who can tell me contractile vacuole is found in which organism? Contractile vacuole is found in which organism? You will tell me, sir, contractile vacuoles are found in the case of amoeba. What is the function? Osmoregulation and excretion. Osmoregulation, excretion are the functions of contractile vacuole. Okay, contractile vacuole. Now, what about food vacuole? Food vacuole is found in the case of paramecium. In the case of paramecium, we can find food vacuole. Okay, food vacuole. Now, again, I'll ask you one more question here. Centrioles. Centrioles, which type of microtubule arrangement do you find? In centrioles, which type of microtubule arrangement do you find? Do you find in the case of centrioles, do we find 9 plus 1, 9 plus 2, 9 plus 3 or 9 plus 0? Which type of arrangement do you find in the case of right, centriole? 9 plus 1, 9 plus 2, 9 plus 3, 9 plus 0. Quick. See, centriole, all of you know. Centriole is 9 plus 0 arrangement. Centrioles is students, tell me again, think again. Use your brain cells and tell me again. 9 plus 0 or 9 plus 2? Ravindra Rani, I saw your answer. I saw many of you telling the wrong answer. Cilia and flagella. Cilia and flagella is 9 plus 2. Again, I'll repeat. Cilia and flagella is 9 plus 2. Centrioles is 9 plus 0. Because in the case of centriole, in the right in the middle, there is something called as hub. Hub formation is there in centriole. Okay. But in the case of cilia and flagella, in the middle, we have 2 other microtubules are present. Okay. So let me ask you one more question now. In the case of cilia and flagella, what do we observe? Tell me the answer now. In cilia and flagella, we observe, students, in cilia and flagella, we observe 9 plus 2 arrangement. Cilia and flagella, we observe 9 plus 2 arrangement. Yes, basal body and centriole is 9 plus 0. Exactly. Sankey. Okay. So 9 plus 2 in the case of where? 9 plus 2 students in the case of your cilia and flagella. Okay. See, inclusion bodies. There are multiple types. Phosphate granules, cyanophysine granules, right? Glycogen granules are multiple types of reserve food material or inclusion bodies. Which of the following statements about inclusion body is going to be incorrect in nature? They are involved in ingestion of food particles. Okay. They lie freely in cytoplasm. Reserve food material. Not bound by any membrane. Yes. So you can see, students, Reserved material in prokaryotes are stored in the cytoplasm. These are not bound by any membrane. They lie free in the cytoplasm. They lie free in the cytoplasm. Free. Oh, should I show the answer by mistake? Show the answer by mistake, students. Okay. Not by any membrane, free, and ingestion of material. So they are involved in ingestion of food? No, they are not involved in ingestion of food. Option number A is the answer. Okay. Which is the important site for formation of glycoprotein and glycolipid in eukaryotic cell? Again, students, Amazon factory. Who is the Amazon factory? Who can tell me, students, who is the Amazon factory here? Quick. Who is the Amazon factory here? Who is the Amazon factory? It's going to be Golgi body. Golgi body main work is packaging. Here, for example, glyco and glyco, it is added by Golgi complex. Added by Golgi complex. While this protein part is from RER. Now, students, can anyone tell me from where do we get this lipid part? This lipid part is from where, students? Who can tell me? Lipid part is from SER. It is from smooth endoplasmic reticulum. Lipids from SER. This is from RER, rough endoplasmic reticulum. That is protein part. Protein part is from rough endoplasmic reticulum. Okay, okay. Done. Done. Next question on the board. Which organelles are included in the endomembrane system? Again, students, endomembrane system is what? Endomembrane system means it's a gang. See, in the school, all of you have a gang. I have a gang. Like I have a boys gang, which from, let's say from 5th standard we are together. Till now, every Sunday we still meet. Even after 20 years or 15 years of our friendship, we still meet. The gang is there. Why? The gang is still able to maintain because all of us are connected. All of us are interconnected. All of us are think alike. All of us think alike because here also endomembrane system, the gang, all the gang members functions are coordinated here. Okay. Now, what is the trick, students? Who can remember the trick? The trick is every gamer loves victory. E for endoplasmic reticulum. Okay. E for endoplasmic reticulum. G for Golgi complex. Golgi complex. Mitochondria comes here. No. Mitochondria is here. No. Mitochondria is here. No. Every gamer loves victory. Every gamer loves victory here. Every gamer loves victory. Exactly. So endoplasmic reticulum, Golgi complex, lysosomes, and vacuole. Very easy, right? Very easy it is. So easy. So easy. So easy. Students, recently I was teaching a class on Science Olympiad. Okay. Recently I was teaching a science class on Science Olympiad, and I told all of them this small fact, and they were shocked. Okay. How many of you know, students, which is the largest organism on Earth? Which is the largest organism on Earth? Quickly, students. Which is the largest organism on Earth? I'll give you a small clue. Students. This organism does not belong to kingdom Animalia. What? Yes. This organism does not belong to kingdom Animalia. Who can tell me which is the largest largest organism on Earth? It is an organism which does not belong to kingdom Animalia at all. Cicovia, not Cicovia also. Not Cicovia. Not blue whale. Not blue whale. It is not blue whale. Who can tell me? So this is how I teach my small kids also, like small kids, not my kids. I don't have kids, I'm not married. Okay, small kids as in junior science Olympiad kids, right? Yes. It's a fungi. Yes. The largest organism on Earth is a fungi. So it is a fungi which is kilometers long. Not 1-2 kilometers. Kilometers long it is. The fungi is kilometers long because of the mycelium it has. And all of you know, mycelium is part of fungus. It is a part of fungus. So it can grow for so long. So long it can grow. Not anaconda. Anaconda, bro. Anaconda. 20 foot is maximum in anaconda also. 20 foot of python. Okay, maximum it can grow 20 feet in anaconda also. Okay. So it's a fungi. Largest organism on Earth is fungi here. Okay. One more question which I asked them recently. All of them were shocked. Okay. All of you can tell the answer very easily. Okay. Tell me, students, which is the closest star to our Earth? This all of you can tell. Which is the closest star? Like you see the sky, you can see the star very clearly. You can see the star. You can see the star so nicely. You can see the star. Which is the closest star to the Earth? Who can tell me? Closest star to the earth? Who can tell me, students? Closest star to the earth? Sun, exactly. Sun is the closest star. By definition, the definition of a star is anything which has its own heat and light. Own heat and light, and sun is the closest star to the earth. Okay, not moon. Moon is a satellite. Who is telling moon? Moon is a satellite, not a star. Moon does not have its own light. Moon reflects the light of the sun. Okay, sun, exactly. Moon, it seems. Okay, next question on the board. Next question on the board, students. Which of the following is incorrect statement? Incorrect statement, students. Which of the following is incorrect statement? Students, GK fail, no? Tell me properly. GK fail, no? GK fail. Tell me yes or no. No, gas is getting a heat from the cylinder, no. Oh God, GK fail. Following is incorrect statement. Perinuclear space forms a barrier between material inside the nucleus and out of the cytoplasm. Yes, makes sense. Nuclear pore acts as a passage for the proteins and RNA molecules to go in and out. Yes, it makes sense. Mature sieve element possess conspecies nucleus. Wrong. Microbiotes are present in both animals and plants. Yes. So what is wrong here, students? In statement number 3, what is wrong? Student, GK is my favorite. Like, I love GK a lot. I don't know why. Okay. Why 3 is wrong, students? Because it says possesses. There is no possession here. Everything is lost. Everything is lost here. Everyone is removed from the house. No one is present inside. It's hollow. Okay. Hollow. Nucleus, no. It's going to be absent. Next question on the board, students. Again, remember, students, it's same for erythrocytes also. Erythrocytes are basically red blood cells. No, again, students, can anyone tell me? Erythrocytes, do they show cell division? Do they show cell division? Tell me yes or no. Erythrocytes, do they show cell division? Who can tell me? My dog is saying no, no, no. Do you agree with Cash? I agree with Cash. I agree with Cash. He's telling no, no, no. So erythrocytes do not show cell division. Why? Because, students, when erythrocytes they are born, right, they go and die eventually. And there is something called as a graveyard. There is something called, students, there is something called as graveyard for red blood cells. Who knows the answer? What is the graveyard of red blood cells called as? Who can tell me? Students, did you also have this story in your school that your school is built on a graveyard? In my school, it was a lot. My seniors used to tell me, our school is built on a graveyard. I don't know why. To scare whom, I don't know. Okay. So, yeah. Spleen, exactly. Spleen. They don't divide. Okay. So, where is Cash, students? Cash is my dog, students. My dog's name is Cash. Imagine name. I hope so. He is a black color Labrador. Okay, he is a black color lab. He is black color, full pitch black. Okay, shiny boy. Okay, I kept his name as Black. I kept his name as Cash. So black money has to come in life. Okay, so yeah. Light green, exactly green. Okay, students. Spleen is there. Next question on the board. Again, students, how many of you get confused? How many of you get confused between cristae and cisternae? Raise your hands. Oops. The pen fell. Students, how many of you know the difference between cristae and cisternae? Who can tell me, students? Who can tell me? Result gotilla, wait ಮಾಡಿ. Yes, students. Okay, tell me, quick. Do you get confused between cristae and cisternae? There's a small trick here. Okay, all of you remember, students, cistae is little small in like when you read the words. This is much more longer. Okay, much more longer here. So how can you remember this? Remember, students, compared to Golgi apparatus or Golgi body and mitochondria, Here, how many words are there? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14. So here we have 14 words. So much more longer here, much more longer here. So here sister name is going to be, sister name is longer. So longer to longer. Simple as that. Simple as that. Okay. Simple as that. If you ever get confused, just remember that. Okay. Just if you get confused, just remember that. Okay. Now, cyste is going to be smaller, so mitochondria, thylakoids, flat membranous structure. Centromere is going to be primary constriction in chromosome. Sorry. Done. What is the answer, students? What is the answer? Option number 1. When centromere is situated in the middle of two equal arms, students, very easy question again. The chromosome is referred to as. What do you call the chromosome? It's present in the center. If centromere is present in the center, what is it called as? Quick answer I want. Does not take a lot of time. It should not take a lot of time. Okay. It should not take a lot of time. Students, smash the like button, all of you. Students, have you smashed the like button first of all? Target is not reached today. Students, at least make your target reached. Okay. It's going to be metacentric in nature. Okay. If the pH in lysosomes is increased to alkaline, what will be the outcome? See, all of you know, students, in lysosomes there are certain enzymes called as hydrolytic enzymes, and all of these hydrolytic enzymes, they function very well. Hydrolytic enzymes will function very well in acidic medium. But if you make it to alkaline medium, it will not work. Okay. So will become inactive. They will become inactive in nature. Students. Okay, they'll become inactive in nature. They cannot function very well. Students. They cannot function very well. They become inactive. Students. Okay. Next question on the board, students. Again, it is a type of your match the following when you talk about your different type of plasters here. Chromoplast will give you carotenoids. Who can do first? Competition. Amylopectin is going to be a starch. Elio oil, and eleuroplast is a protein layer. So eleuroplast is going to be protein, storage of protein. So that is the answer. I got my answer. Students, I got my answer. Do you get your answer? No one is liking, exactly. Everyone is watching, but no one is. Who got the right answer first? Me or you? Tell me. Pramod, option number 2 is the answer. Okay. Now again, students, very easy. Again, we will do first finger first. Okay, let you get the answer first. Membrane-bound organelles for endomembrane system functions are function system coordinated. Mitochondria and chloroplast are not considered part of endomembrane system. Yes, both are correct. Students. Both are correct here. Both are correct. Easy peasy question. Exactly. 528 votes, 339 likes. People are simply voting, not even liking. Simple voting you're doing, not even liking. Next question on the board, students. Again, let's see who can do first. Which of the following statements are correct in context of Golgi apparatus or Golgi body? It is important site for formation of glycoprotein and glycolipids. Correct. It produces cellular energy in the form of ATP. Wrong. It is your energy is not produced. It is mitochondria. It modulates protein synthesized by ribosomes and ER. Correct. Okay. Transport of ions. No. Mechanical support. No. B. ANC. ANC is the answer, students. ANC is the answer. Yes, ANC is the answer, students. ANC is the answer. ANC is the answer. ANC is the answer, students. Done. Next question. Which of the following functions is carried out by cytoskeletonous cell? All of you know cytoskeleton. It could be your microfilaments, it could be your microbodies, it could be intermediate filaments. All of them perform a lot of functions. For example, they give the shape to the cell, they provide mechanical strength. But apart from that, students, they also help in one special function which Everyone tends to forget that. It's a NEET 2023 question. Okay, their main function is also motility. They also help in motility. If you can see, students, the cytoskeleton of a cell are involved in many functions such as your mechanical support, motility, maintenance the shape of the cell. This too you'll remember generally, but motility is generally forgotten. So one word they can pick and give you the question. Okay, one word they can pick you up and give you a question. Very carefully you have to answer. Okay, very carefully you have to answer, students. Okay, match the following. Let's see who does it first, me or you. Competition starts. 3, 2. Don't see the question. 3, 2, 1. Time stop. Time's up. Time starts. Nucleolus site for RNA synthesis. A is going to be 3. Okay, 1 is 1. A is going to be 3. D is going to be 1. I got my answer, students. I got my answer. I got my answer already. I got my answer already. Nucleolus. As soon as I see D is going to call D is going to be 1 and A is going to be 3. There is only one option like that, and B has to be 4, obviously. D is 2. Cartwheel. Leucoplast, storage of nutrients. Option number 4, exactly. Option number 4. Okay. Students, DNA present in chloroplasts. Again, I told you, students, in the case of chloroplast as well as in the case of your prokaryotes, it's double standard DNA. Double standard circular DNA. Double standard circular DNA is present there. In your chloroplast, mitochondria as well as prokaryotes, same type of genetic material is present. Right? Endosymbiotic theory. Circular double standard. Circular double standard. Again, match the following. Time starts now. Let's see who gets it first. Again, this is a NEET 2024 question. Let's see who gets it first now. Time starts now. Axoneme is the core in the part of a ciliary flagella. A is going to be 2. A is going to be 2. Okay, these 2 are gone. Okay. Cartwheel pattern. B is going to be 1. Got my answer, students. Got my answer already. B is going to be 1. C is going to be 4. D is going to be, again, obviously 3 that time. 3. What is the answer, students? What is the answer? What is the answer? What is the answer? What did you get? Quick. See, I told you axoneme or core. Axoneme or core is part of ciliary flagella. Cardial pattern, I told you centrioles. Cis crista is going to be part of mitochondria. Satellite is part of chromosome. What is satellite, students? Satellite is the secondary constriction. Satellite is the secondary constriction part on top of your chromosome. If this is a chromosome right here, on top of it you can see satellite like this. Satellite on top. Not like a moon, different from moon. Okay. Next question. Cell wall formation in bacteria is facilitated by which organelle forms in cell wall in bacteria? See, if it is bacteria already, you cannot have ribosomes, you cannot have centrosome. Both of these options are gone. Now, all of you know, students, ribosome main function is what? Ribosome main function is protein synthesis. So again gone. So the only option left is mesosome. Option is mesosome. Because this bacteria, when I say bacteria, I mean prokaryote. Prokaryotes. And in prokaryotes, we do not have Golgi complex or centrosome. We do not have it. So answer obviously becomes mesosome in such case. Mesosome in such case. Ribosome is protein synthesis. Ribosome is protein synthesis ribosome. Okay. It becomes mesosome, obviously. Okay. This is a NEET 2024 question again on the board. Which of the following represents sister chromatids? Sister chromatids. Okay. See, students, this is centromere. So A is gone. Okay. C is talking about same chromatid only. Gone. Okay. Now what about one from here and one from here? That is non-sister. These two, one from here and one from here. Okay. One from here and one from here, students. That becomes your option number B. That becomes option number B, sister. Okay. That becomes B. Okay. That becomes B. Option number B. Okay. D is non-sister. One from here, one from here. One chromatid from here, and one, that becomes non-sister. D is non-sister. D is non-sister, students. D is non-sister. Okay. Again, students, match the following. Time starts now. NEET 2024 question. Let's see who gets it first again. 3, 2, 1. Time starts. Ciliary flagella, part of your axoneme. endoplasmic reticulum, part of your ribosomes, mitochondria, cristae, kinetochore, spindle fiber. Kinetochore, spindle fiber. Kinetochore is a point where all the spindle fibers will attach. For example, this is the chromosome. Here we have kinetochore, where all the spindle fibers attach here. Done, students? Done. What is the answer? D is going to be 1, is only one option, students. D is going to be 1, there's only one option. Option number 2. Option number 2. Okay. One more question, students, on the board. The Golgi cisternae are concentrically arranged near the nucleus with distinct convex cis or forming phase or concave trans or maturing phase. Again, students, who can tell me the answer? Last question, no? Last question. I was wondering. Last question. The cis and trans phase of organelles are identical and interconnected, students. Are they interconnected? No. They are not interconnected. They may be identical to each other, but so they may be, they are not identical to each other also. So they are not identical. Yes. One is cis phase, one trans phase. They are separate. So this becomes false. This becomes false here. This becomes false. A is true, R is false. Next question. Done, students. Done. Not identical, but yes, not identical. They are not identical. Okay. So with that, the cell chapter is over. Now you realize, students, in cell chapter, majority of the questions are from organelles and functions. So you need to know every single organelle and its function. Okay. Let's, for example, if I ask you a question right now. Okay. Who gave fluid mosaic model? What is your answer, students? Who gave the fluid mosaic model? Answer quickly in the chat. Who gave the fluid mosaic model? You will tell me, sir, Singer and Nicholson. Singer and Nicholson gave the fluid mosaic model. What if I ask you, students, one more question? In the case of your prokaryotes, what is the outermost layer called as when it is very solid, thick and tough? Who can tell me, students? Who can tell me? What is the outer layer of your prokaryote called as when it is thick and tough? Glycocalyx. Thick and tough is capsule. So capsule is the answer that time. Capsule is the answer that time. I ask you one more question. Okay. Tell me, students, what is the cytoplasmic connection called as? Let's say here I have one cell, you have one more plant cell. The cytoplasmic connection is there. What do you call the cytoplasmic connection? The cytoplasmic connection is called as what? Plasmodesmata. It is called as what, students? Plasmodesmatal connections. Plasmodesmatal connections. Okay. Now, students, can anyone name one organelle which is found in your animal cell which does not have any membrane? Which does not have any membrane. Quick, students. One organelle in the case of animal cell which does not have any membrane. Animal cell. It is found only in animal cell. It is found only in animal cell. You can't tell ribosome here because ribosome is found in your plant cell also. The answer has to be centrosome. Centrosome does not have any membrane. Centrosome does not have any membrane, students. Centrosome does not have any membrane. Don't tell ribosome because ribosome is present also in the case of plant cell. Also in the case of plant cell. Okay. I'll ask all of you one more question. Okay. Students, tell me what is the function of centrosome? Not centriole. Centrosome has to be the answer. Not centriole. Centrosome has to be the answer. Centrosome has to be the answer. Okay, centrosome is the answer. Function of centrosome is cell division. It helps in cell division. It helps in cell division. Are we missing anything, students? Are we missing anything? Okay. Students, what is the membrane of vacuole called as? Vacuole has a membrane around it. In the case of plant cell, what do you call that? What do you call that, students? What do you call that? What do you call the membrane of your vacuole in the case of plant cell? Very easy again, very easy. Very easy, students, again. It's called as tonoplast. It is called as what, students? It is called as tonoplast. Tonoplast. Done, students. Done. So can we start biomolecules now, students? Can we start biomolecules? Now, students, again, we'll do biomolecules, cell cycle. Then I'll give you 3 minutes break again. Because before plant physiology, I want all of you to have fresh mind. Before plant physiology, I want all of you to have fresh mind. Okay. Now, students, can we start biomolecules now? I want everyone in the chat again. Everyone in the chat, cell the unit of life, done and dusted in the chat, all of you. Cell the unit of life, done and dusted. And let me drink some water now. Let me drink some water also. And students, quickly smash the like button. All of you. All of you? Yes, cell done. All of you feeling confident in cell now? All of you feeling confidence in cell now? Confidence in cell is there? Tell me yes or no. Yes. Done and dusted, cell done and dusted. Everyone? Exactly, cell is done and dusted now. Now, shall we start with now biomolecules now? Biomolecules again, very easy chapter. Very easy chapter. If you have watched my lecture, recent lecture on biomolecules in Hero to Zero to Hero, okay, very easy it is. Very easy it is. Okay. Now, biomolecules again. First question on the board. Prosthetic group differs from coenzyme in which way? What is the difference between prosthetic group and coenzyme? Prosthetic group and coenzyme. It's a difficult question starting only. Difficult question starting only, students. Okay. Quick. They require metal ions for activity? No. Prosthetic group are tightly bound apoenzyme to the apoenzyme. Okay, makes sense. Their association with apoenzyme is transient. Okay. They can serve as a cofactor number of enzymes. The question is, how does the prosthetic group differ from coenzyme? So if you watch my lecture, right, I've told you in my lecture, students, this is me again here also, you can see my arm and my half of my face. I have told you, students, prosthetic group are tightly bound to a coenzyme. Coenzyme are loosely bound or transly bound. But the question is asking us, how is prosthetic group different from coenzyme? So prosthetic group is different from coenzyme because prosthetic group is tightly bound to the apoenzyme. It is very tightly bound to the apoenzyme. Now, students, to the apoenzyme. So all of you tell the answer. Can tell the answer again here also, students. Again here also. Exactly, hero to zero. Okay. See, students. Again, they can ask you one more question here. They can ask you question based on the example also. See, prosthetic group example is heme group. Heme is the prosthetic group here. Heme is the prosthetic group. All of these are examples of cofactors. Cofactors. And all of you should know cofactor is non-protein part. Non-protein part, without cofactor the enzyme cannot work. Okay. Now example of your coenzyme is NAD and NADPH. Example of metal ion is zinc here. Zinc is an example of metal ion here. So remember these examples. Everything is taken from NCERT only. Everything is taken from NCERT only. Okay. Look at this question, students. Which of the following compound is main constituent of lecithin? What is present in lecithin? Let me give you a small clue. Lecithin is found in the case of your lipid bilayer. In lipid bilayer. What is the answer, students? What is the answer? What is the answer? Is it going to be phosphoprotein or phospholipid, students? Phosphoprotein or phospholipid? It is going to be phospholipid. Exactly, students. Students, how much is your marks in the chat? Who can tell me your marks? Phospholipid. Okay, phospholipid. Next question on the board. Concanavalinae. Again, every single table is very important in your biomolecules. There are two tables. One table for secondary metabolites, one table for all the different types of your proteins, functional proteins. So the question here is concanavalinae is a type of. Concanavalinae is a type of. What is it? What is concanavalinae? A pigment, an alkaloid essential, a lectin. Students, if you know this table, you can tell me, students, concanavalinae is lectins. Now, shall we play a game now? Simple game again. Any table, small game. I'll give you 15 seconds to look at this table and then I'll ask you 2 questions now. Time starts now. Go. 15 seconds. Done, students? Done? Done. Okay. Which type of secondary metabolite is codeine? Codeine is which type of secondary metabolite? Quick answer I want. Codeine, which is used in cough syrup. Codeine, which is used in cough syrup, is a type of which type of secondary metabolite? Codeine. That's the first question. Second question is, students, ricin is which type of secondary metabolite? Ricin and abrin. Ricin is which type of secondary metabolite? Ricin is which type of secondary metabolite again? Ricin also. Ricin is a toxin. Very good. Abrin is a toxin. Then your codeine is a alkaloid. Codeine is a alkaloid, students. Codeine is a alkaloid. Very good. Very good. So vinblastin, students, vinblastin is found in the case of your Vinca rosa plant. And this vinblastin has properties to cure cancer. Students, how many of you know what is Vinca rosa? How many of you know what is Vinca rosa, students? How many of you know what is Vinca rosa? How many of you know what is Vinca rosa? How many of you seen this? How many of you seen this? This is called as Vinca rosa. It comes in white color. It comes in a pink color also. Pink and white color. Vinca rosa. It's called as Vinca rosa. It has something called as vinblastin, right? Vinblastin, which helps in cancer treatment. Okay. Okay. Next question on the board. Let's see who gets the first answer. Okay. Now, students. Which of the following glucose transports is usually insulin dependent? GLUT4 1, 2, 3, 4. Which is it? GLUT4. Exactly. GLUT4. You can see, students, again, this table is very important. This table is very important table. You can see here GLUT4 enabling glucose transport into the cell. Okay. Now here again they can ask you all of these. They can ask you one by one separately. They can ask you all of this. All of this can be asked. Okay. Yes, called periwinkle also. It is normally grown in my house. Yes, they can make a tea. You can make a tea also out of it. Tea. Nice tea you can make. Students, do you know you can make tea out of? I forgot the flower name, students. Leguminaceae plant flower. Leguminaceae plant flower, students. You can make a purple color tea out of it. Purple color tea. I made it long back. It was very bad. I could not drink it, but if you can, you drink it. Okay. Next question on the board. Identify the basic amino acids from the following. Very easy, students. See, it says glycolic acid here only, so gone. Tyrosine, valine is going to be neutral amino acid. Lysine is going to be basic amino acid. So, students, can anyone tell me, students, not hibiscus, it's a leguminaceae plant. Leguminaceae. Not hibiscus is very common. Hibiscus is very common, students. Leguminaceae plant family. The thing is, I remember the families, I don't remember the examples. Leguminaceae, it was leguminaceae only. Or Fabaceae, whatever we call it. Okay. So, lysine is a type of your basic amino acid. Can anyone tell me what are the other examples of basic amino acid? Okay. Yeah. Students, never try. Students, okay. So, how many of you know my boost curd story? How many of you know my boost curd story? Caminalia, I think. Boost curd. So, what used to happen was, students, when I was a kid, my parents used to go to the office, or when they were going out of the town or something, they leave me empty. They leave me without supervision in the house. Without supervision, I used to be in the house. Okay. And what I used to do was, I used to go to kitchen. I used to pretend I'm a chemist. Okay. I know chemistry. So what I used to do was I used to mix random food items and try to find one ultimate food item which no one has invented in the world. So I used to be a food scientist. Okay. Once I tried to mix boost and curd, let me tell you, it tasted really good. If you eat sugar and rice, very good. But I'll tell you the worst combination till now. The worst combination till now, which made me throw up. I 4-5 times I vomited that day. Okay, never try it in your life, students. I'm telling you, never try it. If you mix tomato ketchup and curd, ugh, tomato ketchup and curd, the worst possible combination in the world. I made it once, I did the mistake. You don't do the mistake. Okay, so never do the mistake. Okay, never do the mistake. See, boost rice is still fine. It's one of my good inventions. It is one of my good inventions. Okay, but never try ketchup and curd. Oh my god, that's the most disgusting thing I've ever tried in my life. Okay, my god. And I have a weird taste. I eat so many weird things, but that was the next level weirdness. Okay, next question on the board, students. Next question on the board. Okay. Identify the substance having glycosidic bond and peptide bond respectively in the structure. Glycosidic bond is found where? In the case of your carbohydrates. Peptide bond in the case of your proteins. In the case of protein, students. Idli with toothpaste. Oh, you're brushing idli also now. Who eats toothpaste? It is not like mayonnaise. It is not even like mayonnaise. It is not even like see, mayonnaise and curd people mix. You know, students, all of you know, mayonnaise and curd can be mixed. That's how ranch is made. That's how ranch. All of you know what is ranch. In US and all, they make ranch. You know, they add a lot of dill and everything. That's called ranch making. In ranch, they add mayonnaise and curd. They mix. They mix hung curd and mayonnaise. That is better. But tomato and okay, glycosidic bond is going to be your glycosidic bond in the case of your inulin. Inulin is a polymer of fructose, and then insulin is a protein. Yes, option number 3. Option number 3, students. Option number 3. Okay, all of you students, inulin. Inulin is a type of your polymer of fructose, long chain of polymer fructose. Okay, long chain of polymer of fructose. Monomer of fructose. Which of the following most abundant protein in animal kingdom, animal world? Most abundant protein in the animal world, students. Most abundant protein in animal world. Once again, students, give me a minute. Students, they can ask you one more question here, okay? They can ask you a question saying that which is the right, which is the uh, they can ask you a question, one more question. They can ask you which is the most abundant protein in the whole biosphere. Then what is the answer, students? Tell me what is the answer then? Most abundant protein in the entire biosphere. What is the answer then? Tell me. Yes, Clitoria is the yes. Clitoria is the flower. Then Rubisco is the answer. Then Rubisco is the answer. Rubisco is the most abundant, yes, protein in the entire biosphere. Abundant protein in the whole biosphere. Okay, very cool. Collagen in the case of animal world. Okay. Next question on the board, student. Next question on the board. Following are statements with reference to lipids. Lipids have only single bond. It's called unsaturated. No, unsaturated means double bond. Means double bond. Okay. Lecithin is a phospholipid. Yes. Trihydroxypropane is glycerol. Yes. No. No. What is the answer, students? What is the answer? What is the answer? Username is unknown. I have to check. Even I don't know that. Okay. Even I have to check that. What is the answer, students? Option number A, BNC only. Okay. BNC only is the answer. Okay. Next question on the board, students. Identify the incorrect pair. Lectins concatenate drugs, ricin. Ricin is a toxin, so that is incorrect already. Alkaloids is codeine. Yes, toxin is arbutin is a toxin. Ricin is also a toxin. Ricin is also a toxin, students. Ricin is also a toxin. Ricin also is a toxin. Okay. Now again, students, this entire table you should know. Very important table. Okay. Next question on the board, students. Match the following. Let's see who can do it first. Protein will have peptide bond. A is going to be 4. A is going to be 4. This is gone. This is gone. A is going to be 4. Unsaturated fatty acids will have double bond. B is going to be 1. B is going to be 1. C is going to be 2. Nucleic acids is going to be phosphodiester bond. Yes. Phospholipids will have glycosidic bond. So answer is 3. Answer is C, students. Answer is 3. You have seen me teaching botany here. I always teach botany only, students. What is this? 90% time I teach botany. Okay, students. 80% I teach botany. 10% I teach zoology. 5% I teach chemistry. 5% again somewhere down the line. I have taught social. I have taught even English grammar. I teach a lot of English grammar also. Those of you know, I have taught a lot of English grammar also. For your 12th standard students, for your 10th standard students, English grammar also I teach. 10th social also I teach. Okay, what to do? I have taught recently. I have taught history. I have taught geography, and 1 chapter of civics. I have taught 10th chemistry recently. Okay, everything is there. Next question on the board. Which of the following is not a secondary metabolite, students? Very easy question. If you know the table, you know the answer. Curcumin and vinplastin is, rubber is, morphine and codeine is. Amino acids and glucose are not a secondary metabolite. They are not a secondary metabolite, students. They are not a secondary metabolite. Okay. Mixed subject. No option, students. No option. Teach everything. And I was a sports teacher also. The funny part is I started with a sports teacher. Funny part is, students, I started as a sports teacher. Can you believe that? Students? I started as a sports teacher, and then rest everything went on. Oh God. Very easy again. Okay. Now next question again. In the enzymes, which catalyst is broken down? Again, students, this is hydrogen peroxide. Hydrogen peroxide breaking down into H2O plus water. I told you starting with prosthetic group 1. Example, remember the prosthetic group. All of you remember, students, prosthetic group. The prosthetic group, what is it? I used to get, when I used to teach sports, I used to get thorns like sports teacher speaking good English. That was a very rare thing they were telling me. Sports teacher with good English. Sports teacher with good English. Okay, students, again, quick to me the answer, students. Very easy. It is prosthetic group. I told you the first table I showed you. The heme. Heme was the answer, students. Heme is the prosthetic group. Heme is the prosthetic group there. Okay. Now, students, who can tell me what about zinc? Where do we get zinc? Zinc is part of metal ion. What about NAD? NAD for coenzyme. NAD for coenzyme. Okay, NAD for coenzyme. Okay. NAD for coenzyme. heme. Again, the same table, same thing follows here. Very easy. Okay, very easy. Okay, students, very easy question, students. All of you know the basic rule. ATGC. Now here it says adenine is 30%, adenine is 30%, cytosine is going to be 20%. So both are going to be equal. Both are going to be equal. Tell me, students, they're asking you what is the percentage of thymine and guanine. Thymine is going to be 30%, guanine is going to be 20%, 30 and 20. 30 and 20 is the answer. Lepton. 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 Lol, what is a lepton? Okay. What is the answer, students? What is the answer? Option number 2. Okay, lepton. Okay, next question on the board, students. If it is visible to all of you, well and good. If it is not visible, I will tell it orally. Okay. Amino acids have a property of ionizable nature of NH2 and COOH groups, hence have different structure, different pH. I told you. Amino acids are just like all of you. You behave differently in front of different people. When no one is there in the house, you're dancing in the house, watching TV, playing games, eating on top of the sofa only. But as soon as the dad is coming to the house, what do you do? As soon as the dad is coming to the house, what do you do, students? Tell me. Okay. If dad is coming to your house, what do you do? Open the book. Even if the book is ulta, you don't care. Just book should be in your hand. Look, not even looking here and there. Go and wear one, hide all the belt in the house. Okay. And try to start reading out of nowhere. Doesn't matter what you're reading, you will start reading. Similarly, they will act differently. So correct here. Now, amino acids can exist as zwitterion, acidic and basic pH, students. No. The thing is, zwitterion will only exist. Zwitterion will only exist where? It will exist as zwitterion at isoelectric pH, not at acidic, not at basic. At a isoelectric pH will exist. Okay. Okay, students. So at isoelectric pH will exist. So option number 2 is going to be wrong here. So what is the answer? Oh my God, what is the answer, students? What is the answer? Yes, what is the answer, students? One is right, 2 is wrong. Okay, isoelectric pH, exactly. Okay, students, match the following. Let's see who does it first. Match the following. Let's see who does it first. Yes, neutral pH. Isoelectric pH is a pH where I understand what happens in isoelectric pH. In isoelectric pH, what is happening is there is something called as intramolecular proton transfer is happening. Intramolecular proton transfer, when it happens, you get a zwitterion. Zwitterion has something called as a partial positive charge and a negative charge at the same time. So net charge is 0 in your in the case of your zwitterion. Students, I didn't even start solving, you told the answer. Adenine is a purine, A is going to be 4. Okay. Anthocyanin is a pigment, B is going to be 1. Okay. Chitin is a polysaccharide. Okay. And codeine is going to be an alkaloid. So D is going to be 3. Now again, a question to all of you students. Is chitin a homopolysaccharide or heteropolysaccharide? If you tell this wrong answer, gone case. Gone case. If you tell the answer wrong, gone case, students. Footstep is my ear is very sharp. Friends, what I did? When dad used to have a bike called a Super Splendor. Okay. So when I gave the bike to the servicing, I made sure dad's bike gets a different type of horn. You can modify the horns, you know that, right? So when I gave the bike to the garage for servicing, I told the mechanic to change the horn only. And I told my dad the horn was gone, so we had to change a new horn. So as soon as the dad enters, obviously he has to horn somewhere when he was parking inside the building is coming. So the horn used to be played and the horn used to be different sound. That's how I got to know if dad is coming or not. Next level hacks were there. Okay. It's a homopolysaccharide, not hetero. Homopolysaccharide. Don't tell wrong. It's a Students, this is what I don't like in all of you. How many times I've told you, students? Oh my God. Now, again, I want everyone to stare at this. Every single time I teach this chapter, every single time I teach this chapter, all of you still tell it wrong. I don't know why. Where is the stupid polysaccharides, friends? Polysaccharides. Every single time I teach this concept, all of you tell it wrong. Students, read. I want everyone to read this. I want everyone to read this from here to here. Read this much. Everyone read this much, students. All of you read. No. I have told this so many times. I've told this, students. Every single time, all of you make the same mistake over and over. All of you read this. Quick. Exoskeleton of arthropods, for example, have complex polysaccharide of chitin. These complex polysaccharides are mostly homopolysaccharide. There were students. They're homopolysaccharides. They're homopolysaccharides. So all of you will remember now from now onwards. Tell me yes or no. All of you will remember, students. Yes or no? Tell me, students. Please don't make this mistake again and again. I have seen this mistake happening so many times at this point. So many times this mistake has happened. Okay. So don't make this mistake again. Done? Done, students? All of you remember? You have to remember. Okay. Now, read the question again, all of you. Read the question. Very easy question on the board. Very easy question on the board. In a reaction catalyzed by enzyme, which of the following statements is correct? Following statement is correct here. Enzyme decrease the activation energy for formation of transition state. Yes, correct answer. The enzyme make transition from substrate to more difficult. No. Enzyme increase the activation energy. No. Enzyme substrate complex formed during the reactions last for very long time. No. So all of you should know, students, by now. This is called as activation energy. This is called as your activation energy. What is activation energy, students? Activation energy is the energy which is energy difference between your transition state and substrate. The average energy difference between both of them is called as activation energy. Now, what is an enzyme doing? Enzyme is basically reducing the activation energy. Can you see? It is reducing the activation energy. That is the main function of an enzyme. Main function of an enzyme is to reduce the activation energy. Now, cellulose is a polymeric polysaccharide. Okay. The building blocks of cellulose are glucose molecules. Quick answer, students. Quick answer. Very easy question. Doesn't require much thinking here. Students. Doesn't require much thinking. Quick. Polymeric cellulose is polysaccharide. Yes, they are polysaccharides. Building blocks of cellulose are glucose molecules. Yes, they are monomers of glucose. They are joined together. Students. Okay. Next question, students. Username, I have thought that in my Biomolecules chapter. In Biomolecules chapter, when I was teaching, I have thought it that time. What exactly happens in every single scenario? Okay. Read the following statements on lipids and find out the correct set of statements. Okay. Lecithin is found in plasma membrane of is a glycoprotein. So glycolipid. Wrong. It is a phospholipid. Saturated fatty acids possess one or more double bonds. No. Saturated will have single bonds. Gingerly oil has lower melting point, hence remains winter. Yes. Lipids are generally insoluble in water but soluble in organic solvents. Yes. When fatty acids esterified glycerol, it forms monoglyceride is formed. Yes. So CDE. CDE is the answer. CDE is the answer. Okay, students. Done. CDE is the answer. Karthik, Usha. Students, how many of you? I hope your data is not over already, or battery is not over already, because we have a lot to go still. We have a lot to go. Okay. Now, students. So remember, students, gingerly oil. Okay. Again, easy question on the board. Again, easy question on the board, students. Easy question on the board. Exoskeleton of arthropods is composed of. Exoskeleton of arthropods is composed of, students. NEET 2022 question. Okay. Exoskeleton of arthropods is composed of. It is cutin, no, friends. So, do you see the confusion here? The confusion. They have deliberately put something called as cutin here. Okay. So when you're in hurry, what do? When you're in a hurry, like Usha in hurry right now, Usha also in hurry, what they'll do is sometimes instead of reading the question properly, becoming overexcited, you'll go for cutin. Like, oh, cutin, huh? Nice. Sounds like chitin only, but you don't see the spelling that time. So you go for cutin instead of chitin. So don't make that mistake again in examination. Read the question properly because NTA sometimes can be like a snake. They can give you some difficult questions in the middle. Okay. Next question on the board. Which of the following is not a secondary metabolite? Not a secondary metabolite. Curcumin, morphine, lectin, anthocyanin. No, anthocyanin is not a secondary metabolite. Okay. Anthocyanin is not a secondary metabolite. Exactly. Cutin. Done? Anthocyanin is not a secondary metabolite. Oh, students, I read as lectin. Oh my god, I was deceived right now. It's lecithin. Students, I'm going blind right now. I'm going blind. Okay, lecithin. Oh my god, I read as lectin. I read it as lectin. I am only going back. So NTA 1, Basavaraj Sir 0 right now. Oh my god. After spoiling something, I'm going blind now. Lecithin. Next question on the board, students. Inulin is a polymer of. Oh god. I'm also going blind now. Yes. I read it as amylopectin. I don't know why. I read it as amylopectin. Inulin is a polymer of fructose. Malonate inhibits growth of pathogenic bacteria by inhibiting the activity of, students, competitive inhibitor. Competitive inhibitor, students, competitive inhibitor. Which of the following is a competitive inhibitor? Which of the following is a competitive inhibitor, students? Succinate dehydrogenase. Succinate dehydrogenase, students. See, inhibition of succinate dehydrogenase by malonate, which closely resembles the substrate succinate in nature. Next question. Cellulose does not form blue color iodine because cellulose cannot hold. It cannot hold iodine because it does not have complex helicals. Does not have complex helicals. It cannot hold. Yes, I'm going chapter wise. Students, I'm sitting down for 2 minutes because my leg is hurting a lot. I don't want leg to swell a lot. I have to keep the swelling to minimum. Cannot hold I2 molecules. Okay, students, with that your biomolecules chapter is also over. Okay, again students, in biomolecules you should only focus on biomolecules, mainly focus on tables. You will notice that many questions are from the tables, the table for secondary metabolites as well as table for your proteins. Okay, so students, shall we start with cell cycle and cell division now? Shall we start with cell cycle and cell division? Yes. So with that, biomolecules is also done and dusted. With that, biomolecules is also done and dusted right now. Okay, biomolecules is also done and dusted. Okay, now we come to cell cycle now. Again, students, cell cycle is a very high scoring chapter, because every single question, students, every single question from cell cycle will come from where? It will come from the Meiosis 1. If you know what exactly happens in your leptotin, zygotin, packaging, diplotin, diakinesis, half the work is over. So I'll give you a small revision beforehand. Listen to me. In the case of leptotin, what happens? Chromosome become gradually visible under light microscope. Zygotin, what happens? In the case of zygotin, what happens, students? In zygotin, synaptonemal complex formation happens and synapses happens and bivalent and tetrad formation happens. Now, what happens in packaging? In the case of packaging, there is formation of recombination nodules, recombinase enzyme and crossing over. And bivalent and tetrad becomes clearly visible, packaging. Okay. Then we have diplotin, students. In diplotins, what happens? Chiasmata formation happens, synaptonemal complex disappears. In diakinesis, in the case of diakinesis, what happens? Termination of chiasmata. Then they can ask you question was, when does the centromere split? Centromere splits in anaphase of mitosis, anaphase 2 of meiosis. That much you remember, students. That much you remember. Okay, that much you remember, students. Okay. Students, after this I'll give you a break. After this chapter I'll give you a break, students. After cell cycle I'll give you a break. Before we start the plant physiology, I'll give you a break of 15 minutes. Okay, so I just want to rest my legs. I don't want legs to become very swollen because the doctor has told me do not stand for more than 2 hours. Now we are doing the class from 10 to 11, 11 to 12, 12 to 1. From 3 years I'm standing, right? So doctor has told me not to stand more than 1.5 hours actually. Okay, so I will just try to put some cream on top of it and apply a bandage and come in after the break. Okay, so let's start the cell cycle and cell division, students. Let's start with cell cycle and cell division right now. Here's the first question on the board. Students. Spindle fiber attached to kinetochore of the chromosome during. Again, if this is the chromosome right here, we can see spindle fiber attaching, and all of this will happen where? In the case of metaphase. In the case of students, metaphase. In the case of metaphase, it happens. In metaphase, it will come and join. In the case of metaphase, students, it will come and join. In the case of metaphase, so please remember, very easy it is. Very, very easy it is. In the case of metaphase, it happens. Okay. Metaphase, it happens. Next question on the board, students. It's a simple match the following, and it's a NEET 2024 question. This is how you can expect the question again this year also. Okay. See, diakinase, I told you. What happens in the case of diakinase, students? Termination of chiasmata, I told you. Packaging, what happens? Packaging, appearance of recombination modules. Zygotin, what happens, students? Synaptonemal complex formation. Termination of chiasmata. Okay. Zygotin, formation. Leptotin, chromosome look like thin threads right now because starting stage, starting stage. Because the starting stage. Okay. So what is the answer, students? Tell me what is the answer. A is going to be 2. Only one option is there. Students, only one option is there. Only one option is there, students. Okay. Next question you see. Following are the stages of cell cycle. You're supposed to tell me which are the stages from cell cycle. Okay. Like let's say if G1 is first, then we have S, that is synthesis. Then we have G2. G1 is first. Then we have G2. Then we have synthesis. Then we have G2. Then we have chirokinesis. Then we have cytokinesis. Cytokinesis at the very end. Okay. Tell me what is the answer. Students, tell me what is the answer now. Yes, students, I'm from Karnataka. Okay. So answer is option number 3. Option number 3 is the answer. Next question, students. Chromosome becomes gradually visible under light microscope during leptotin stages. Makes sense. The beginning of diplotin, okay, diplotin stage, right, is recognized by dissolution of synaptonemal complex. Correct. Synaptonemal complex disappears. Then there is a formation of chiasmata. Formation of chiasmata also happens in the case of diplotin. Okay. Both are correct here. Diplotin, what happens? Disappears and X-shaped mark called as chiasmata formation happens. Okay, next question, students. Again, you will see a trend in NEET 2024. A same trend is going to be there in this year also. They'll ask you match the following. Very easy, you can tell me. Very easy answer. Free marks, all this is free marks. Okay. Chromosome starts pairing together to form synaptonymal complex. Again, I told you, synaptonymal complex is going to be zygotine. Okay. Chromosome visible under light microscope. That is going to be leptotine, first stage. 4 chromatids of bivalent chromosomes becomes distinct and recombination nodule appears. That is going to be pachytene. Students, they become clearly visible, and as well as recombination nodules, crossing over, recombinase enzyme, everything is pachytene. Dissolution of synaptonymal complex, that is going to be diplotine again, diplotine. So tell me the answer. Tell me the answer, students. Only bio PYQs, bro. Only bio. I'm doing only bio PYQs. Only bio PYQs, students. Only bio PYQs. Tell the answer, student. Tell the answer now. Answer is option number 4. Okay, option number 4 is the answer. Students, are you able to solve the questions? Tell me yes or no in the chat. Are you able to solve the questions? Because you can see a pattern here. Again, I'll repeat one more time before we go ahead. First stage, right? In the case of leptotine, what happens? Leptotine chromosome becomes gradually visible under light microscope. Zygotine, formation of synaptonymal complex, synapses, bivalent or tetrad formation. Next stage, pachytene. Pachytene stage, what happens? In the case of pachytene, bivalent becomes more clearly visible. Recombination nodules, crossing over, as well as recombinase enzyme. Then diplotine. Diplotine, what happens? Chiasmata formation, synaptonymal complex disappears. Last stage, diakinesis, termination of chiasmata, and every other organelle disappears. Okay. Next question on the board. Now, what happens in G1S, G2? See, all of you know, students, if you are confused, all of you can tell me, students, G0 is going to be 1. C is going to be 1. Tell me the option where C is going to be 1. This is gone, this is gone. Now, you also know, students, S phase, what happens? Replication of DNA happens. Okay, replication of DNA. So, S is going to be what? 2. It's going to be 2. Now, A is going to be what? 4. Cell is metabolically active and grows continuously. And C is going to be 1. And D is going to be 3. Since of protein. So, if you know 2 options, you can tell the entire answer. You can tell the entire answer if you know 2. That is the easy method of, entire match the following. So, match the following is actually very easy. Match the following is actually very easy. Okay. So, match the following is actually very easy in such way. Very easy. No. Crossing over. See again, student. As soon as you say crossing over, pachytene should come to your mind. Pachytene should come to your mind, students. Pachytene should come to your mind as soon as you say crossing over. Okay. Between which chromatid in which stage of the cell? All of you know it happens in your pachytene stage. Okay. Pachytene stage, it happens of prophase 1. Okay. Now, non-cystic chromatid, homologous chromosomes, yes. It is supposed to be between homologous chromosome. It has to be between homologous chromosomes. Pachytene, non-cystic chromatids, homologous chromosomes at pachytene stage of prophase 1. Prophase 1. Very easy, no? Very easy. So, it's questions from your cell cycle. I will call all these questions as Laddu questions, free questions, rank boosting questions. Rank boosting questions, all of them are. Rank boosting questions. Okay. You again see these questions. It's very easy. Tell me the answer in the chat. I'll give you 10 seconds. I'll give you 10 seconds to solve this question. 10 seconds. G1S, G2. Right. G1S, G2M. G1S, G2. Okay. Very easy. Rank booster question. Cells in G0 are. What are these, students? They're temporarily suspended. They are suspended in cell cycle. They are temporarily suspended in cell cycle. They cannot divide. They do not divide. They just exit the cell cycle after G1. After G1, they exit. Okay. They exit. Okay. Next question on the board. During meiosis 1, which stage synapse takes place? Again, students, I told you, synapse where it happens. Tell me, students, second stage, no? Second stage, zygotine synapse happens. Along with synapses, there is formation of synaptonymal complex happens. Along with that also, synaptonymal complex formation also happens. Okay, so very easy, done. Next question on the board. Attachment of spindle fiber to kinetochore chromosome becomes evident in. Okay. When does it become more evident in? It happens in the case of metaphase. Again, same metaphase, kinetochore attachment happens, spindle fiber attachment to the kinetochore. Okay. Dissolution of synaptomal complex. Dissolution of synaptomal complex occurs where? It occurs in the diplotin stage. In diplotin stage, it happens. Along with that, what happens, students? Along with that diplotin, what happens? Tell me. Formation of chiasmata. Formation of chiasmata. So, this cell cycle chapter, the concepts will repeat year after year, year after year. Same questions will repeat year after year. Again, see this 2020. Identify the correct statement for with regarding G1 phase or gap 1 phase. Reorganization of cell of all components takes place. Cell is metabolically active but grows but does not replicate DNA, students. Yes, it grows, does not replicate DNA. Does not replicate DNA. DNA replication happens where? DNA replication happens in the S phase. It happens in the S phase. Now, which stage of meiotic prophase shows termination of chiasmata diakinesis? That's all. Diakinesis. Termination of chiasmata diakinesis. It is that easy. It is that easy, students. And all of this is only possible. All of this is only possible if you're well versed. If you do not remember anything, again, very difficult for all of you. Now, see this question. Now, the centrioles duplicate under where? Centrioles duplicate, students, under your S phase. Now, here, if I were your examiner, I'll make the question a little difficult now. How can you make it difficult, students? I can make it difficult by saying that centrioles duplicate in which phase and inside dash. Okay, inside. Is it nucleus or cytoplasm? Nucleus, cytoplasm, student, tell me. Inside the nucleus or inside the cytoplasm? You will tell me, sir, inside the cytoplasm. Inside the cytoplasm it replicates. So that's how question can be made a little difficult. Okay. See here. In the nucleus, DNA replication happens. Centrioles duplicate in the cytoplasm. No. Which of the following stages of meiosis division of centromere happens? Division of centromere, all of you know, students, centromere is splitting only in 2 stages. In mitosis, anaphase. Meiosis where? Anaphase 2. Why? Because anaphase 2 is very similar to mitosis anaphase. Both are same actually. Okay. So it is going to be anaphase 2. So if you get a question in examination, in which stages of mitosis and meiosis, centromere splits, the answer should be what, students? The answer should be anaphase of mitosis and anaphase 2 of meiosis. This should be your answer. Student. This should be your answer. Okay, this should be your answer right now. Clear, students? Clear? Now, bivalent tetrad formation is characteristic feature of observed in. Bivalent tetrad is present in the case of zygotine. Okay. In the case of zygotine. Now, if I change the question, students, bivalent tetrad formation is characteristic feature clearly observed in. Clearly or more visible. Then what is the answer, students? Tell me what is the answer that time? Tell me. If it is more clearly visible, then it becomes pachytene. If it is pachytene, what is the answer, students? If it is pachytene, what is the answer? There's no option only, but pachytene. It should be pachytene that time. So you have to be very careful on what your question is asking you. More clearly visible or less clearly visible under your this thing also. Okay. Clear, students? Not lipidine, it's going to be pachytene. Geeta Shu, I can't say the name, students. I'm blind. I have to get my glasses. I don't know where I kept my glasses. So it's supposed to be pachytene. It cannot be 3 because synaptomal complex is not in your Packaging. It cannot be 3, that's why Christina. It cannot be 3 because it is not packet synaptic complex is forming where? It is forming in zygotine. So you can't write this. Okay, you can't write this. Now, in meiosis, crossing over a genetic material between homologous chromosome catalyzed by the enzyme. Which enzyme, students? Which enzyme is present? Who can tell me? In packaging stage, which enzyme is present? Ribozyme is present. We have the ribozyme. So, recombinase enzyme. Okay. Recombinase. Brain is not breathing now. Ribozyme is your RNA enzyme. Recombinase enzyme. Okay, recombinase enzyme. Students, all these questions are so same. Appearance of recombination nodules. Again, students, you know the answer already. Appearance of recombination nodules in homologous chromosomes during meiosis is characterized by. Wherever there is recombination nodule, it happens at the site of crossing over. Whenever there is going to be a crossing over, it occurs there. Right? It occurs there. Right? Yes, it occurs there where the packaging happens in the packaging stage. Students, do you believe or not? Tell me when I say cell cycle is the easiest chapter. Cell cycle is the easiest chapter. All of you believe me or not? Tell me. See here. During G0 phase of cell cycle, the cells is metabolically inactive. Is it inactive, students? Tell me yes or no. The centrosome undergo duplication during S phase of interphase. Yes. Again, it is going to be incorrect because I told you it is going to be metabolically active, not inactive. Metabolically active it is going to be. Metabolically active it is going to be. And done, students. Done. With that, your entire cell cycle is done. I know cell cycle is so easy. Cell cycle is all direct questions. You don't have to think a lot. If you know what exactly happens in what stage, you can easily get the marks there without any tension. But again, I'm telling you, if you don't revise now, examination you will have very less time. So the whole logic is that the more time you spend now on these chapters, less time you spend in examination. How many of you understood? Students, the more time you spend now on these chapters like cell cycle, morphology and all, the more time you spend on them now, right now, the less time you'll spend in examination. In examination, you don't have to think again, oh, packaging, what happens? Where does the synaptic complex happens? Where does the, recombination nodule happens? Where does the termination of chiasmate happens? No. You don't have to think even twice then in examination because you have done thinking right now. Okay, so with that, cell cycle is also done. We'll start with photosynthesis in higher plants after a small break. All of you go eat food and come. All of you go eat food and come. We will start the class at 2:15 or 2:20. Or 2:15 only is fine. What's the time right now? My watch is 15 minutes, 10 minutes faster. Okay. All of you eat and come. Okay. All of you quickly go eat food, aram se relax for some time. Okay. And then come back and then we'll finish the plant physiology, which all of you have doubts in. So I don't want any of the students to miss plant physiology, especially students. Plant physiology is going to be very, very important. If you don't want to miss, you have to come back. Okay. Okay, students. So I'll just go rest my leg for a while. I'll apply some cream and apply some crepe bandage over it. And I'll also eat something, right? Eat breakfast. If you did not eat breakfast. Okay. Eat and come back and we'll start exactly at your 12:15. Until then, students, all of you, I want everyone to finish the target of 500 likes. At least finish 500 likes by then. Okay. So bye bye, students. Until then, take care. I'll come back and I'll see you all of you at 2:15. Hello students, welcome back. All of you ate something and came? Tell me yes or no in the chat quickly. All of you ate something? Yes? Students, give me a minute. Let me just set this up. The PDF was cracking a little. Yes? Hello students, hello, hello, hello. All of you ate? Nicely ready? Last stretch. All of you ready for last stretch now, students? Yes, done. Downloading is done. Perfect, perfect. All of you ready for the last stretch? Yes, welcome, welcome, welcome. Now, students, we come to the last stretch of the today's class 11th. Right, there are mainly 3 more chapters left. All 3 chapters are very high weightage, especially the first 2 chapters of your photosynthesis in higher plants as well as respiration in plants. These 2 chapters hold a lots of weightage, and every single year, right, you get innovative questions from here. And I'm very sure many of you feel like, sir, this is very, very difficult to solve. Don't worry, you have my notes. Cyclic, non-cyclic photophosphorylation, Calvin cycle, you have my notes. C4 cycle, you have my notes, right? Then we come to respiration plants, you have my notes for your entire glycolysis, Krebs cycle, etc. Everything is present with you. Everything is present with you, students. Everything is present with you. So you don't have to worry about anything in that regard, right? Just try to solve some questions. I'll give you some idea which type of question can come, from where the question can come. Just chill and solve the questions now. Okay. So let's start, students. Let's start with the last part of the chapter, last part of your class 11th botany. That is going to be photosynthesis in higher plants. Very easy, very, very easy. Okay. Now, first question on the board, students. Okay, first question on the board. One scientist cultured cladophora. In a suspension of azotobacter and illuminated the culture by splitting the light through prism. He observed that bacteria accumulated mainly in the region of. This was the experiment done by a scientist called as T.W. Engelmann experiment. In T.W. Engelmann experiment, students, he got to know that aerobic bacteria is accumulating in the 2 regions. For example, if this is your cladophora here, in all the aerobic bacteria was accumulating in this one region here, and remaining aerobic bacteria was accumulating in one more region. Why, students? Because whenever we take a prism and we split the light, when we split the light here, right, when we split the light, we get Vibgyor. And especially, students, especially in the red region as well as your blue region, that is a favorite to the entire plants. They like to eat on it, like, oh, light, they'll go very crazy about it, right? So it is going to be red and blue and red light. Blue and red light are the T.W. Engelmann experiment, according to T.W. Engelmann experiment, favorite regions of the plants. Okay. Next question on the board. In hatch and slack pathway, that is basically your C4 pathway, C4 cycle pathway, the primary CO2 acceptor is. Who is the primary CO2 acceptor in the case of C4 pathway? When the CO2 is coming, who is doing namaskara? Welcome to the club. Welcome to the C4 cycle. Who is doing that, students? Who is doing that? Who can tell me, students? Who can tell me? Quick. Who is the first primary CO2 acceptor, students? If you don't remember, let me show you. Sure. This is your entire C4 cycle. Here you can notice, students, when CO2 is entering, the first acceptor is PEP. What is PEP, students? PEP is nothing but phosphoenol pyruvate. Phosphoenol pyruvate is the primary acceptor of your CO2. Now, if I ask you a question, which is the first stable product here? The first stable product is going to be, where is it? OAA, oxaloacetic acid, which is 4 carbon. That is why it's called a C4 cycle also. That is why it's called as C4 cycle. The first stable product, oxaloacetic acid, is the first stable product here. So the first acceptor is PP phosphoenol pyruvate. Now I can change the question. Who is the primary CO2 acceptor in the case of PCR cycle? PCR cycle. What is the answer now? Tell me, students. What is the answer now? PCR cycle, also called as Calvin cycle, also called as C3 cycle. Who is the primary CO2 acceptor here? The primary CO2 acceptor here is none other than RUBP. It is none other than RUBP here. Rubulose 1,5 bisphosphate is the primary CO2 acceptor in the case of C3 or C4 or anything. RUBP is the answer that time. RUBP is the answer. Okay, not Rubisco. Rubisco is the enzyme there. Rubisco is the enzyme. Okay. Next question on the board, students. During non-cyclic photophosphorylation, when the electrons are lost from the reaction center of PS2, what is the source which is replaced the electrons? For example, in case of non-cyclic, there is a continuous loss of electrons. Someone is accepting the electrons, but who is accepting the electrons? Who is the Karana here? Who is donating the electrons? Take more, take more, take more. The continuous supply of electron is coming because of photolysis of water. That is called a splitting of water. Yes, splitting of water. It's going to be splitting of water. Okay. You can see, students, from a non-cyclic, when the electrons are going up 680 nanometer, here water is being split open and it is releasing electrons and protons. It's been given here. So it's going to be what is the answer? What is the answer here? Next question on the board, students. The oxygenation activity of Rubisco enzyme in photorespiration leads to the formation of what exactly are the product of your photorespiration? All of you know there are two products. One is PG, one is PGA. PG is 2 carbon, PGA is 3 carbon. What is PG? Phosphoglycolate. PG is phosphoglycerate. So what do we get, students? What do we get? 1 mole of 6 carbon, no. 4 carbon molecule, no. 2 moles of 3 carbon, no. We get 1 molecule of 3 carbon plus, the answer should have been 1 molecule of 3 carbon plus 1 molecule of 2 carbon. This is the full answer. This is the full answer, but it is not given here a full answer. Okay. This is supposed to be a full answer here. See here, students, what exactly happens here? When RUBP, which is 5 carbon, oxygen, how many carbon, students? Oxygen is 0 carbon. So the product has to be 5 carbon only. See, oxygen is 0 carbon. Unlike CO2, it could have given you 6 carbon in total. But here, 5 carbon has to be transferred here. So what do we get? We get PGA plus PG. And PG is 2 carbon, this is 3 carbon. But in the option, they have not given you this part. This option, they have not given you phosphoglycolate. They have only given this. So the most likely the answer is, the most probable answer is going to be option number 1. That is going to be 1 molecule of your 3 carbon compound, that is PGA, 3 carbon compound. So, but the full answer has to be what, students? The full answer has to be 1 molecule of 3 carbon and 1 molecule of 2 carbon. This was supposed to be the full answer. All of you understood, students? All of you understood? Tell me yes or no in the chat quickly. Tell me yes or no in the chat. All of you understood why the first answer is right? All of you in the chat, why the first answer is right? All of you understood? Chintu, Pramod, Christina, Meghana, all of you understood why the first answer is correct? Yes. So please be clear on what is exactly photorespiration also. Next question on the board. In light reaction, okay, this is a very top quality difficult question. Okay. In light reaction, plastoquinone facilitates the transfer of electrons from. Plastoquinone is present here, PQ, CY, PC. Here, primary electron acceptor, that is your pheophytin, right? Pheophytin. Here, electrons are coming from where? Electrons are coming from PS2. From PS2, electrons are transferred here. So PQ is between what? PQ is between your PS2 and cytochrome B6F complex. So PQ is present between this and this. This is, they have not mentioned this in the examples anywhere. So you can just write PS2 to cytochrome B6F complex. It cannot be from here to here because PQ is present here. It cannot be this. Okay. It cannot be this. So PS2 to cytochrome B6F complex. So I'll just show you one more time, students. Where is that this thing? Here, you can see it here clearly one more time. So from PS2 it is coming up. Pheophylline is going to be a primary electron acceptor. Here we have PQ, CY, PC. PQ is present where? PQ is present between CY and P2. So all of you can get an idea. All of you can get an idea here. Okay. Next question on the board. Which of the following systems is if statement is incorrect statement? Grana lamellae has both. Grana lamellae. Grana, yes, grana has both. Cyclic photophosphorylation involves. Oh, students know. Cyclic does not involve both. Cyclic will have only PS1. Only PS1 in the case of cyclic. PS1, PS2 both involved where? Both are involved in non-cyclic. Both are involved where? In the non-cyclic, but in cyclic we only have PS1. We only have PS1 in the case of cyclic. Understood all of you? Understood by option number 2? Tell me yes or no. Understood by option number 2? Yes? All of you students quickly smash that like button, all of you. 500 likes is not done still. Huh? Students, please smash the like button. Next question on the board. The first stable product of your CO2 fixation in sorghum is sorghum. Sorghum follows C4 pathway. C4 pathway. Okay. So they're asking you which is the first stable product in your C4 pathway. Right? C4 pathway. Which is the first stable product in your C4 pathway? Which is 4 carbon molecule. All of you know it is OAA. OAA is the first stable product in your C4 cycle. So option number 4 is the answer. Okay. This is PGA. PGA present in your C3. Phosphorylaceric acid is present in C3. 2 is not the answer. 4 is the answer. OAA is the first stable product in the case of C4 cycle. Now, following crops have been extensively cultivated in CO2 rich environment highly for. Because students, here you learn about C3 plants. 2 C3 plants which are cultivated very extensively. 2 C3 plants. Which are those, students? Who can tell me? Tomatoes and bell pepper. Tomatoes and bell pepper. Tomatoes. Who says tomatoes? Tomato. Okay. The products of light reaction are what are produced in light reaction? 2022 question. In light reaction, do we get water? No. Water, no. Do we get CO2? No. Do we get ATP? Do we get NADPH? Do we get oxygen? Yes. We get all the following. In light reaction, we get all the following. Water is being not produced, it is being consumed. Water is being consumed here. CO2 is not produced here. CO2 is not produced in the case of your starting stages. CO2 is not produced. Plants take the CO2 in C3. Plants take in the CO2 in C3 cycle. That is dark cycle. Okay, dark reaction. Here the question is, what is produced in light reaction? ATP, NADPH, both are produced where? Both are produced in your non-cyclic. Oxygen is also released in your non-cyclic. Clear all of you? Clear? Yes or no? All of you understanding? Should I repeat again? Should I explain again? Tell me yes or no in the chat quickly. I'll explain again. Do you want me to explain the question, this question again one more time? Tell me yes or no in the chat quickly. Do you want me to explain this one more time? Yes or no? You want me to explain? See this. In the case of your cyclic, you can see easily, students. In the case of your cyclic light reaction, right? We get ATP here, we get ATP. Do we get NADH? We get ATP here, we get NADH here, and we get oxygen here. Somewhere oxygen has to come. It's not here. Oxygen is also here. So oxygen is released, NADPH is released, ATP is released. But is there water is being released? Water is not being released here. Water is being consumed here. Water is being consumed here. Is CO2 released anywhere? No. CO2 is not released anywhere. CO2 is consumed in dark reaction. CO2 is consumed in your dark reaction. Okay. Now, next question. The primary CO2 acceptor in C4 plant is phosphoenol pyruvate, PEP, phosphoenol pyruvate, and is found in mesophyll cell. It is found, students, in the mesophyll cell above. Okay. Mesophyll cell in C4 plants lack Rubisco enzyme. Yes, because Rubisco enzyme is present in bundle sheet. Rubis coenzyme is present in your bundle sheet, students. Okay, Rubis coenzyme is present in bundle sheet. Clear, all of you? Clear? Yes or no? Tell me, students, in the chat quickly. Very simple it is. In mesophyll cell, students, what should I explain you one more time? See here we have something called mesophyll cell, bundle sheet cell. In mesophyll cell, what happens? Primary CO2 fixation happens. Pepcase is the enzyme present here. At the same time, in the case of bundle sheet, what do we have? We have the C3 cycle happening here, and Rubis coenzyme is present here. Rubis coenzyme is absent in mesophyll on top. Done? Yes. So both are correct here. Okay, both are correct here, students. Okay, both are correct here. Now, next question on the board. Which of the following is not true regarding the release of energy during ATP synthesis through chemiosmotic hypothesis? Not true. That's not true. Okay. Reduction of NAD to NADPH on the stroma side of the membrane, yes. Breakdown of proton gradient happens. Electron gradient is not involved here. You do not have any type of electron gradient in the case of chemiosmotic. Chemiosmotic is entire proton gradient. There is no use of electron gradient here. There is no breaking of electron gradient here. So, not true is option number C. Next question. Which of the following combinations is required for chemiosmotic hypothesis? There are 4 main requirements. All of you know there are 4 main requirements for chemiosmotic hypothesis. 4 main requirements. Who can tell? Which are the 4 main requirements? Quick. Students, quickly smash the like button, all of you. All of you quickly smash the like button first of all and mark your attendance. Done? See, this proton pump is required, electron gradient not required. NADPH synthesis not required, electron gradient not required. Intact membrane, proton pump, proton gradient, and ADP synthesis. Option number 2 is right. Students, answer should be mistake. How many ATPs and NADH are required for synthesis of 1 molecule of glucose? Students, before we go to 1 molecule of glucose, I want everyone to tell me in the chat how much of ATP and NADPH is used in one cycle of Kelvin cycle. Who can tell me in one C3 cycle how many ATP and how many NADPH is used in one cycle? If you know in one cycle, students, what happens is in every one cycle there is 3 ATP is being used and 2 NADH is used. And NADH is used here. And there's 3 ATP also. 2 are used in reduction, 1 is used in regeneration. Now this is for 1 CO2. For 1 CO2 this is used. Now the question is asking per glucose. So for glucose, how many carbon we have? 6 carbon. Right? C6H12O6, 6 carbon. So this cycle has to go 6 times now. For one cycle, 2, 3, 4, 5, 6. 6 times it will happen for 1 glucose. So what do we get, students? 6 into 3 ATP as well as 6 into 2 NADH. What do we get, students? 18 ATP as well as 12 NADH. 12 NADH is found here. 12 NADH is found here. So 18 and 12 is the answer. 18 and 12 is the answer right now. Okay, 18 and 12 is the answer. Done? Done. Next question. The reaction center of PS2 has an absorption maximum at. What is the maximum absorption at PS2, students? PS2, what is the maximum absorption? PS2, 2 is bigger, 700, huh? Students, is it 700? 2 is bigger, so 700. Tell me yes or no. Since 2 is bigger, it's going to be 700? No. It's going to be, 2 is going to be 680 nanometer. What about 1, students? 1 at 700 nanometer. Okay. Students, that is the end of photosynthesis higher plants. I did not take a lot of questions because photosynthesis, what happens is, questions will repeat all the year. If you look at 2024 also, same question is repeating every year. Okay. Now we come to respiration in plants. Okay. Now here, before we go to respiration, let me tell you what is most important in photosynthesis. In photosynthesis, everyone should know what is Calvin cycle. Everyone should know what is C4 cycle. Everyone should know, I think some questions are missing, students. Some question will come later on. Everyone should know what is going to be a light reaction, cyclic and non-cyclic. 90% of questions will come from here only. Remaining questions will come from what is the reaction center, what are different pigments present. They can ask you what is different experiments they can ask you. That's all. Okay. I think some questions are coming after this respiration plants. So right now, students, photosynthesis higher plants are done and dusted. Very easy it is. Nothing difficult in the chapter. Okay. So all you need to do is revise the photosynthesis higher plants once theory. If you watch, you'll get to know. So all you need to do, students, if you have any confusion right now, right, all you need to do is just type photosynthesis in higher plants in 3D on YouTube. My video will come. Watch the video once. Okay. So photosynthesis in higher plants in 3D you can watch easily. Photosynthesis in higher plants in 3D. Now RIP starts. Okay, RIP. First question on the board. Where is the respiratory ETC electron transport chain located in plants? Do not tell mitochondria. They're asking you where inside mitochondria. Inside mitochondria where? Outer membrane, inner membrane, intermembrane. Where is the entire thing present here? Okay. Yes, this session is only for botany. Do you realize? Botany just class 11th. I am not even doing class 12th botany today. Just botany class 11th I am doing. Imagine. It is taking almost 5 hours we are going. Right. That is the important. That's how detailed we are going. Quickly students, pick answer I want. See, if you remember this structure right here, if you remember my diagram here, okay, here you can clearly see in the inner mitochondrial membrane. In inner mitochondrial membrane, the entire thing is present. The entire ETC is present right here. Entire ETC is present right here. Entire ETC present right here. So what is the answer, students? What is the answer? Inner mitochondrial membrane. Now, if I ask you a question, students, where proton is being accumulated, then what is the answer? Tell me, students. Then what is the answer? Who can tell me? In which region is the protons being accumulated? Who can tell me, students? Quickly. Proton is being accumulated where? Plant physiology, I'll do. One shot where I'll do. Don't worry. I'll do once. Plant physiology, one shot lecture. Tell me the answer to this question now. Where are the protons being accumulated? If you see again this diagram, all the protons are getting accumulated here, in this region here, below region, and this region is intermembrane. So in the intermembrane space, all the protons are being accumulated. Intermembrane. Okay. Now I'll ask you one more question. Okay. Where ATP synthesis is occurring? In which region of your mitochondria ATP synthesis is occurring? We tell ATP happens, but where is it happening? Where is the ATP synthesis happening? ATP synthesis happening where, students? Entire ATP synthesis happening in the matrix region. In the matrix region, again this protons get transferred, and ATP formation happens here. ADP to ATP formation. ADP to APT formation happens in the matrix itself. In the matrix. So you have to be very careful what exactly happens here. Matrix what happens, intermembrane what happens, inner membrane what happens, intermembrane what happens. So be careful on what they're asking you. They can ask you any of the three. Okay. If they ask you, sir, where is the final electrons being accepted? Where is the, see, all of you know the final electron acceptor is oxygen. Final electron acceptor is going to be your oxygen here, half O2. Oxygen here, right? Final electron acceptor. But which location? Again matrix. Matrix of mitochondria. Next question on the board. The respiratory quotient of value of tripalmitin is a lipid. Tripalmitin RQ is what? Tripalmitin, what is RQ? Students, tripalmitin, what is IQ? Who can tell me? Tripalmitin, what is IQ? Quick. This is a direct question, student. Direct question. Doesn't go wrong here. 0.7, very easy. Students, what is the RQ for carbohydrates? in the chat. What is the RQ for carbohydrates? Quick. What is the RQ value for carbohydrates? Who can tell me? Respiratory quotient for carbohydrates. You will tell me 1. What about for proteins? Who can tell me for proteins now? What is the respiratory quotient for proteins? 0.9. Good. Next question on the board. The conversion of glucose to glucose 6-phosphate, the first irreversible reaction is catalyzed by. Now again students, in the glycolysis, what they can ask you, I'll tell you. There is a slide also. Glycolysis, they can ask you the first enzyme, last enzyme, rate determining step, which step water is removed, which step oxidation happens, which step ATP is consumed, which step ATP is formed. All of these they can ask you for the entire, right, for the entire glycolysis. The question here is, the first enzyme is going to be your, who can tell me students? Hexokinase. Beautiful. Hexokinase. Now, number of substrate level phosphorylation in 1 ton of citric acid cycle. In one citric acid cycle right here, in one citric acid cycle, how many substrate level is happening? Who can tell me students? How many substrate level is happening? You can see students, there is only one location where substrate level phosphorylation is happening, and that is right here. Right here, this GTP formation. This GTP formation right here is the substrate level phosphorylation right here. Now if they ask you one more question, in 1 turn of citric acid cycle, how many times oxidative decarboxylation is happening? What is the answer? In 1 turn of citric acid cycle, how many times, right, oxidative decarboxylation is happening? How many times oxidative decarboxylation is happening? Tell me, who can tell me? You should tell me, sir, 2 times it is happening, 2 times. First time it is happening here, right here, first OD, isotrate to alpha keto to succinyl coenzyme is second one. So 2 times oxidative decarboxylation is happening. Similarly, they can ask you students. Similarly, they can ask you. Similarly, they can ask students. Similarly, they can ask you how many times, let's say, CO2 is being released. If they ask you one more time, how many times CO2 is being released? Again, answer is 2 times. 2 times CO2 is being released. Okay. Now, I will change the question, students. I will change the question. How many times CO2 is released for 1 glucose? How many times CO2 is released for 1 glucose? Or how many times oxidative decarboxylation is happening per glucose? Per glucose, how many times happening? Per glucose, how many times oxidative decarboxylation is occurring in 1 Krebs cycle? In 1 Krebs cycle, for 1 glucose, how many times happening? For per glucose, it is going to be 4 times. Per glucose, it is going to be 4 times. You will ask me why, sir? Why? Because 1 glucose, students, 1 glucose will give you how many pyruvate? 1 glucose will give you 2 pyruvate. 1 glucose will give you 2 pyruvates. 1 glucose, 2 pyruvates. Now, 2 pyruvate means? 2 pyruvate means 2 acetyl-CoA. 2 acetyl-CoA means cycle is happening 2 times. So, for 1 glucose, the cycle will happen how many times? For 1 glucose, cycle will happen 2 times. In 1 time 2 CO2, 2 times is 4 CO2. 2 times means 4 CO2. All of you understood? All of you understood, students? Tell me yes or no in the chat quickly. All of you understood, students? Yes or no? Yes or no, students? Yes? No. 1 second, students. 1 second. Done? Yes. Look at this question, students. Which of the following statement is incorrect? They're asking you which of the following statement is incorrect. I hope it is visible. If it's not visible, I will read for all of you. ATP is synthesized through complex 5, correct? Oxidation reduction reaction produces proton gradient in respiration. During aerobic respiration, role of oxygen is limited to terminal role. Yes, in the end, oxygen is coming in taking up the electrons, final electron acceptor. Okay. Now in ETC, electron transport chain, one molecule of NADH gives 2 ATP. No, it gives 3 ATP. 3 ATP. One FADH is going to give you 2 ATP. 2 ATP. So wrong statement. Option number 4 is the answer. 4 is the answer. Look at this question, students. The 5-carbon compound in TCA cycle is. The 5-carbon compound in TCA cycle is an entire TCA cycle. Which is the 5-carbon compound? 5-carbon compound. It starts with something called as alpha. It starts with something called as alpha, students. Alpha. 5-carbon compound. The only 5-carbon compound in your entire cycle. In the entire citric acid or TCA cycle. That is going to be alpha ketoglutarate. The number of times decarboxylation of isocitrate occurs during single TCA cycle. The question is asking you number of times decarboxylation of isocitrate is happening. Now students, to answer this question, all of you should know where is isocitrate coming. Where is isocitrate coming up? If you can see, students, isocitrate is coming right here. Okay, so number of decarboxylation 1 2 do 2 decarboxylation. Okay, 2 decarboxylation. Now students, if I change the question again to, let's say, how many times, see, look at alpha, where is alpha ketoglutarate? Okay, if I change the question, students, if I change the questions to this one to alpha ketoglutarate, then what is the answer? Tell me, students. Then tell me what is the answer. If I change the question to alpha ketoglutarate, how many times it is happening? Tell me. Then it is only once. If it is alpha keto, it is only once. If it is going to be isocitrate, it is going to be 2 times. Okay, so isocitrate 2 times and alpha keto only once. All of you understood? Students, tell me yes or no in the chat. Tell me yes or no, quickly, students. Tell me yes or no. See here. If it is starting from here, 1 happens here, 1 happens here. It starts from here, 1 happens here. Now, if I change the question to succinate, then what is how many? If I change the question to succinate, how many oxide decarboxylation is happening? If we start from succinate, how many is happening? 0. Succinate is 0. Understanding, students? The question is telling you, students, how many times oxidative decarboxylation is happening if the TCA cycle we are starting from isocitrate. If you're starting from isocitrate, how many? If you're starting from isocitrate, 1, 2 is happening. 2 times it is happening if you're starting from isocitrate. If you're starting from alpha keto, then only once it is happening. Only once. If you're starting from succinyl coenzyme A, then 0. Simple as that. See here. The number of times decarboxylation of isocitrate occurs. Isocitrate happens 2 times. Decarboxylation isocitrate is happening 2 times. Tell me yes or no. The number of times decarboxylation of isocitrate occurs during single TCA. Read the question carefully. Where is isocitrate starting from? After this, how many decarboxylation happening? 1 decarboxylation, 2nd decarboxylation. If you're starting from isocitrate. Starting from alpha keto, this is over now. If not, consider this now. Starting from alpha keto, only once. Starting from succinyl coenzyme A, 0 decarboxylation. That is always the concept. Understood, all of you? Understood? Tell me yes or no in the chat. Very easy, right? Very easy. Students, it's very easy. Chapter is very easy. It may look difficult and start from outside, scary from outside, but very, very easy. Okay. Identify the cytochrome which act as a mobile carrier for transfer of electrons between complex and complex 4. Mobile carrier, students. Which is the mobile carrier? Which is the mobile carrier? Quick. Cytochrome C. Very good. Cytochrome C. Okay. Fatty acids are considered connected with the respiratory pathway through. Where are the fatty acids connected? All of you know, students, all of you should know this. Fatty acids, if you see here, again, this diagram is given you in NCERT. If it's not clearly visible, open your NCERT right now. We can see, students, fatty acids are entering through where? Entering through acetyl-CoA. Entering through acetyl-CoA right here. Fatty acids, acetyl-CoA. He's also agreeing. Okay, so acetyl-CoA is the answer. Acetyl-CoA is the answer through which it's entering. Through which it's entering. Okay. Okay. The photochemical phase of photosynthesis, again, photosynthesis includes. Photochemical phase means what, students? Photochemical phase means light reaction. Then what is dark reaction called as? Dark reaction is called as what, students? Dark reaction is called as biosynthetic phase. Photochemical is light reaction. Biosynthetic is dark reaction. So in light reaction, what happens? Release of oxygen, yes. Formation of NADH, yes. Use of ATP, no. Water splitting, yes. Carboxylation, no. ABD. Done? He's calling, I know. ABD, done. Because, see, there is no use of ATP. There is formation of ATP is happening, yes. I can agree with that. Formation of ATP will happen, right? But usage of ATP not happens. ATP is not used in the case of a light reaction. NEET 2024 question. This is so wow. Didn't even see that coming. Okay. In C3 plants, some O2 binds with rubisco, hence CO2 fixation is reduced. Yes, makes sense. In C4 plants, mesophyll cells show very little photorespiration. No. In C4 plants, there is no any type of photorespiration is not found. Photorespiration is completely absent here. Photorespiration is completely absent in the case of your dark reaction. So what is the answer, students? What is the answer? Tell me. Students, tell me the answer quickly. Option number B is the answer, because this is going to be wrong. Because in C4 plants, there is no way we can get photorespiration. Photorespiration is completely absent in the case of C4 plants. Completely absent. Okay. Again, good question again. C4 pathway, students, what happens? Phosphoenol, carbon molecule, phosphoenol. Okay. Light reaction, all of you know, in light reaction we have entire thing. Yes. Photorespiration, what happens? Phosphoglycolate happens. Phosphoglycolate. Calvin cycle, we have RUPP. Light reaction. What is the answer, students? A is 3, A is going to be 3, B is going to be 1, C is going to be 4, D is going to be 2. A is going to be 3, D is going to be 2. Got it. Got the answer. Students. Got the answer. For a second, I was like, where is the option? Okay. For a second, I was like, where is the option? Okay. For a second, I was like, where is the option? Confused. Okay. Okay, students, very easy question, students. How many molecules of ATP and NADH are required respectively to fix every CO2 molecule entering Calvin cycle? 2024 question. All of you know there is going to be how many ATP? 3 ATP. 3 ATP and 2 NADH. 3 ATP and 2 NADH. All of you know this. 3 ATP and 2 NADH. Here is a Kelvin cycle. Here you can see 2 ATP here, 1 ATP here, 2 NADH here only. Okay. Synthesis of ATP is linked to development of proton gradient across the membrane is called as chemiosmotic hypothesis. Chemiosmotic hypothesis explains how the ATP is formed. It explains how the ATP formation takes place. Students. Okay. Which of the following are required for dark reaction? It means they should be involved in dark reaction, for the formation of dark reaction. Okay. Quick. Do we need light? We don't need light. Chlorophyll? We don't need chlorophyll. Okay, we don't need these two. Do we need CO2? Yes. ATP? Yes. NADPH? Yes. We need all the three. We need all the three for your dark reaction. We need all the three for dark reaction. Need all the three for dark reaction. Okay. Okay. Chintu, all of you taking some of the wrong answers. See, for dark reaction, we need CO2. CO2 is to enter and bond with RUBP or PEP in the case of your C3 or in the case of C4. It has to bind. It has to bind, students. It has to bind. There is no other option. So we need CO2. Students, all these are other questions from your which can be asked in your entire Krebs cycle. Why is it called as EMP pathway? Because of three scientists, Emden, Mayer of Parnas, right? Which step water is being split? Which step ATP is consumed 1 and 3? Which step ATP is produced 7 and 10? Rate determining step, third step, irreversible steps 1, 3 and 10. Oxidation happening at 6th step. First enzyme hexokinase, last enzyme pyruvate kinase. So it happens in aerobes and anaerobes, it happens in both. It happens in your, in the case of, it happens in the case of your prokaryotes, eukaryotes, everywhere. Doesn't matter. Glycolysis is the basic step in respiration which happens in your pro-eu everywhere. Okay. Target done, students. Oof, finally. I thought target will not be over. Okay. So with that, students, since the target is over, we come to the last chapter of your entire class 11th. Shall we start, students? Shall we start? Last chapter of your class 11th. All of you. Respiration done and dusted, all of you in the chat. RIP done and dusted. RIP done and dusted in the chat, all of you. RIP done and dusted in the chat, all of you. Done. Now we start with the last chapter of your syllabus, that is going to be your entire plant growth and development. Again, this chapter revolves around examples. If you know example, this is very easy. Not if you don't study examples, very difficult for you. So if you want to make this easy, learn the examples. If you don't want to make it easy, ignore the examples. Up to you. Up to you, students. Shall we start? Yes, RIP done. Everyone in the chat. RIP done and dusted. RIP done and dusted. RIP done and dusted. RIP done and dusted, students. Last chapter. Shall we go, students? Shall we start? Yes. Shall we start? Last chapter. With same energy, same enthusiasm as we had 5 hours ago for the first chapter. Same thing will continue in the last chapter with a bang now. Let's go, students. Let's go. First question on the board. Hey, what is this? Why is this question coming here, students? 1 second, students. This is a question for your... So I'll give you this. Do this in homework question. Why is this question coming here? Lol. Full mood on is spoiled. Students, some questions are mixed up. Oh, it's coming here properly. Okay. In order to increase sugarcane crop, which of the following plant growth regulators should be spread for your yielding sugarcane crop? Like increasing the stem length. Increase the stem length, you should always know gibberellins. Gibberellins used to increase the stem length. So option number 3. Option number 3. Next question. Removal of shoot tip is a very useful technique to boost the production of tea leaves. It is because... Quick. If you remove the shoot tip, what happens? Effect of auxin is removed and growth of lateral is enhanced. You might have seen, students, in northeast, especially northeast, or also in your Assam and in Bengal also it might be there. There's a tea plantation. In tea plantation, you might have seen thick bushy. It'll be very big thick bushy will be there. So what happens if you remove the top part? It will grow laterally. It will start to grow laterally that time. Okay, start to go laterally that time. Okay, laterally. So it is good for tea leaves. Good for tea leaves. Students, if it's not visible, read it for you. It takes a very long time for pineapple plant to produce flowers. Which combination of hormones can be applied to artificially induce flowering in pineapple plant throughout the year to increase the yield? To increase the yield, which of the Ooty also, exactly. Ooty also. Students, Ooty is so beautiful. Ooty. It's raining in Ooty apparently right now. It's raining in Ooty right now. Okay. Now, throughout the year, which of the following spray can be used? Oxalin, ethylene, students. Oxalin, ethylene, all of you know, flowering has to be pineapple. Pineapple means oxalin, ethylene. Again, direct question. Don't have to even think. Okay. Now, students, let's do a small revision before we solve questions. because I feel some of you might have don't remember. Let's do a small revision for all of you before you solve the questions. Okay. Now, just remember this following thing. I'll give you a small revision and then we'll go. Okay. Now, see this. Development effect, or if you have auxin, all of you know, flower initiation, we just now learned, pineapple, parthenocarpy, prevention of abscission. This prevention of abscission is only for young. In young plants, it will prevent. But if the plant is old, then there is no prevention at all. If it is old plant, like go die. If it is old plant, auxin doesn't care for old plants. It is let going old plants. So just remember, in old plants, there is no type of your prevention. Young plants, there is a prevention. Okay, apical dominance, feminizing effect, cell division, cell enlargement. Okay, in gibberellins, understand gibberellins, you should remember this one, something called GA3, the first isolated one. GA3 first isolated. Here, stimulates stem growth. All of you learned in sugarcane example. Promote seed germination. It is promoting seed germination. Bolting rosette plants, for example, all of you know what is rosette. Cauliflower and all is there, no? On nice stem lap. So there will be increase in stem. Rosette plants, delaying senescence. All of this can be asked as a question now. All of this can also be asked as a question now, students. Okay. Now, increases length of grape stalk, increase length of stem, thus increasing the yield. See, sugarcane, increase the yield as much as 20 tons. Elongate and improve shape of apple, speed up the malting process in brewing industry, spraying juvenile conifers with GA hastens the maturity period. Okay. Now, in the case of your cytokinins, what does it do? Discovery is going to be your Skoog and co-workers. Isolation, C.O. Miller. Okay. Cytokinins. Okay. Regulates cell division, modifies. Students, it's not visible, I guess. It's not visible. I don't think it's visible, students. I'll give you this PDF. I'll just read it out now. Just remember. Delays senescence, modifies apical dominance, nutrient mobilization, promote chloroplast development, helps in adding new leaves. Okay. Ethylene, there are multiple effects. Seedlings, helps in seed dormancy, break seed dormancy. Same as gibberellins. Shoot, promotes root growth, root hair formation, ethylene. Induce flowering in pineapple and mango, promotes female flowering cucumber. Ethophone, that is ripening of fruits. Senescence, depoterized plants, where did it go? This is very important, very important. Internode petiole elongation, deep water rice plants. So every single time you might think that, when I say internode elongation, you'll go for gibberellins. You don't go for gibberellins, read what is given. If it says deep water rice plants, go for ethylene in such case. Now, what about the last? Abscisic acid. Promotes seed dormancy. Gibberellin was promotes seed germination, dormancy. Stimulates the closure of stomata. Antagonist to gibberellins, like fighting against gibberellin. Okay. General plant growth inhibitor. Increases tolerance to stress. Initiator of plant metabolism. Seed development and nutrition. Maturation can happen here. Okay. It's also called as stress hormone. Also called as what, students? Stress hormone. Now you can hear, students, you can see here everything is given here. Auxin all functions. Gibberellins all functions right here. Cytokinins all functions. Ethylene all functions right here. And abscisic acid all functions. So you all have to do is remember all of this table here. Remember this table and you're good to go. Okay. Now let's look at the questions. No. Let me answer, students. Sugarcane is going to be, this is gibberellins done. Oh, understand. Plants follow different pathway response to environmental stress, that is plasticity. Plasticity is when plants take a different pathway. Different pathway they take. That is called plasticity. Okay. Here examples, who can tell me the examples of plasticity, students? Who can tell me the examples of plasticity? There is one on environmental and phase also. There is buttercup and then there is something called as cotton, coriander and larkspur. Cotton, coriander, larkspur are the other examples of plasticity. Cotton, coriander and larkspur and buttercup. Buttercup is also one more example. Okay, buttercup is also an example here. The plant hormone used to destroy weeds is 2,4-D. It's a type of your auxin. Here. The phenomena by which undivided parenchyma cells start to divide mitotically during plant tissue culture is called as not dividing cells. They're regaining the ability to divide. Regain the ability to divide is de-differentiation. So something is not dividing. It is regaining divide to again. That is de-differentiation. Okay. After it will stop again. That is redifferentiation after later on. So differentiation, de-differentiation, redifferentiation. Okay. 3 steps are there. Which of the following growth regulators is adenine derivative? Very good question again. Very good question. If you attended my class backlog series on plant growth regulators, there I've taught you this. There I've taught this adenine derivative. Following is the adenine derivative. Very easy, students. Which of the following is the adenine derivative? Cytokinins. Cytokinins are the adenine derivative, students. Again, I have given you those notes. I've given you backlog series notes. I've given you. Refer those notes. Every single hormone you can finish in half an hour. Half an hour you can finish the entire hormones. Okay. Which hormone is used to induce immediate stomatal closure and relief? Stress hormone, students. Which is the stress hormone? Which is the stress hormone? That is going to be abscisic acid. So immediate closing of stomata helps in immediate closing of stomata. Okay. The gaseous plant growth regulator is used in plants. The gaseous plant growth regulator is used in plants too. Gaseous one, ethylene, right? Kill dicot weeds, no, that is for your auxin. Speed of the malting process, that is going to be gibberellins. See students, if you see only root growth, you will go for auxin. But here you can see root hair formation. Root hair formation. Then you have to go for ethylene. Here. Root hair formation, ethylene. Very good. A phenomena which influence auxin and also play major role in discovery. The phenomena where plant is growing towards the light, it is bending towards the light. That led to discovery of your auxin. Who can tell me what is it? Phototropism. Very good. Phototropism. So this is very small. Ignore this question. I can't even see. Students, again these questions are coming. Where is the glycolysis? Suddenly from nowhere, glycolysis questions are coming. Glycolysis. Students, make these your homework questions. Very easy again. There I've told you, in which step ATP is produced, in which step ATP is consumed. Same questions only. Energy yielding process. Which step ATP is produced? Okay. Look at students. Spraying of which of the following phytohormones on juvenile conifers? Spraying juvenile conifers in hastening maturity period. That is going to be gibberellic acid. Okay. Gibberellic acid hasten the juvenile conifers. See this question now. You will think, if you read this question, promotes internode or petiole elongation deep rice plants. If you read only this much, you will obviously go for GA3. But the question says deep rice plants. In such case, you should go for ethylene. So this is a small confusion which can happen. But don't worry, all of you know now. Deep rice plants, ethylene. If there is nothing, if there is just internode elongation, then GA3. Okay. No. But no damage is caused to the grass as auxin does not affect the monocots. Auxin does not affect the monocots. Formation of interfacillar cambium, students, from a fully developed parenchyma cell is called as parenchyma cell starts to undergo dedifferentiation. Parenchyma cell, which is the primary permanent cell starts to divide again. That is called as dedifferentiation. Spraying sugarcane crop which following plant regulators increase the length of the stem. That is again gibberellins, sugarcane crop. Last question for today. Take your time. and to the last question, and we're done with that. With that, your entire botany is over. Ethylene, students. Ethylene, what does it do? All of you know gibberellins increase the stem length. Gibberellins increase the stem length. Promote synthesis and abscission. Delaying leaf senescence. Inhibiting seed germination. Increasing the stem length. D is 1. D is 1. And with that, chapter is over. With that, chapter is over. Okay. Priya, I have explained entire dedifferentiation, redifferentiation in the plant growth regulators chapter. So all you need to do is go to the channel and just type plant growth regulators backlog series. You will get a video. Specifically go to this one point where I'm explaining redifferentiation, because I've drawn a flow chart there. I've drawn an entire flow chart there and I explained. It'll take you hardly 10 minutes to understand the concept. 10 minutes to understand the concept of differentiation, redifferentiation and dedifferentiation. 10 minutes. So just spend 10 minutes and learn the concept from that video. Just type plant growth regulators backlog series in the channel. It is already available. Okay. And students, with that, the entire session comes to an end. Entire session comes to an end, students. So that's it, student. That's it. Okay. That's it, student. That's it. The entire session is over. So I want everyone to comment one thing. See, we can comment so many things. But I want you to comment one thing. Hashtag class 11th botany done. So everyone in the comment section, students, hashtag class 11th bot done. That's all. Or botany done. Okay. So with that, this chapter is also done and dusted. Everything is done at one go. So mnemonics, I have told you mnemonics today when I was teaching you. I told you mnemonics. If you want, in my Telegram, students, in my Telegram, all of you know my Telegram, right? Telegram is in this Telegram. I will add a separate link today. That link to that video which no one has. I will send that link here. So done that. Watch that lecture for all the mnemonics. Okay, students. Done, students. So with that, I am done here, students. Again, if you're a NEET 2026 2025 aspirant, this video is very helpful to all of you. And I wish all of you do not waste any time. Right? I hope all of you have gotten a mock test. I hope all of you are solving mock test. All the students who have bought the mock test AITS, all of you know how well I made the questions there. All of you know how well I have made the questions in AITS. All of you solving those questions, I know. So if you're not bought AITS, students, as soon as possible, get the all-in test series, which will help you to boost your marks. I am very confident on that, because when you sit and solve questions, you become better every single time. And today's session is a proof of that. So if you solve more questions, you become better and better and better. Okay, so solve more questions, become better, become the best version of yourself, and go to the exam. Do not have any regrets. Right now, because there's no point. Once you go to the examination, you should never get a thought in your head that I wish I could have done one more mock test. I wish I could have done 2 more mock tests. That regret should not come to your mind right now or in the examination. So you still have a chance. You still have a couple of days, some 2-3 weeks left. So get a mock test, start solving mock test aggressively, and all the best, students. Thank you so much, students. I will see you next lecture. Bye bye, take care. Do not forget to comment in today's video, and if you want a part 2. Okay, so bye bye, students. Take care, all of you.